Welcome back to The Deep Dive, the show where we slice through complex subjects, sift through the sources, and give you the absolute core of what you need to master. Today, we're embarking on an incredible journey into a topic that is both as ancient as humanity itself, yet, surprisingly, still revealing its deepest secrets: the science of wound healing. Think about it, from our earliest ancestors scraping a knee to modern surgical advancements. Mending our bodies has always been fundamental. Yet despite incredible strides, the exact, intricate mechanisms are continuously surprising us.
It's truly remarkable, isn't it? For something so fundamental to our very survival, the body's repair system is an astonishingly complex, meticulously choreographed ballet of cells, signals, and structural components. Our mission today isn't just about, covering basic first aid. We're diving deep into the intricate cellular and molecular orchestration that dictates precisely how your body repairs itself after an injury. We want you to feel like you've really grasped this, like an elite professional in the field.
That's right. Our source material for this deep dive is comprehensive. A real exploration of the latest understanding of tissue repair, regeneration, and all the myriad factors that can either make or break the healing process. So whether you're prepping for a critical meeting, catching up on the cutting edge of medicine, or just someone inherently curious about the profound capabilities of your own body, this deep dive is your shortcut. By the end of this, you'll have a professional level understanding of wound healing, from its basic biology to the most advanced cutting-edge therapies. Get ready for some serious aha moments that will truly elevate your grasp of this topic. Okay, let's unpack this with some fundamental concepts, because it's important to distinguish between two key terms: wound repair versus regeneration. They sound similar, and we often use them interchangeably in everyday language, but biologically they are profoundly different.
Well, what's fascinating here is how the body prioritizes in most everyday injury scenarios. When we talk about wound repair, we're describing the body's practical, often urgent attempt to restore essential integrity. Think of it like a quick, efficient patch-up job. The primary goal is speed and functionality, resealing fluid barriers, quickly reestablishing blood flow, and ensuring the injured area can function again. This process often and usually results in a scar. A scar, in essence, is a less than perfect reconstruction, prioritizing function and speed over flawless restoration. It's the body saying, let's get this working again fast.
So it's like a functional fix, right, but not always aesthetically perfect.
Exactly.
But then there's regeneration, which sounds like the ultimate healing dream. What's that about?
Precisely. Regeneration is the gold standard, the biological ideal. It's the perfect restoration of the original tissue architecture, and critically, it occurs without any scar formation. Imagine a cut that heals so flawlessly you literally cannot tell it was ever there. That's regeneration. However, this remarkable ability is incredibly limited in adult humans. We mostly see true regeneration during embryonic development in some lower organisms like salamanders that can regrow entire limbs.
Right, like the classic salamander tale.
Exactly. Or in very specific human tissues such as bone and the liver, which possess unique regenerative capacities. For most adult human tissues, particularly skin, it's typically wound repair, not regeneration, that occurs. That's the stark contrast and the big challenge we face.
So even with all our incredible medical advancements, achieving perfect scarless healing for most injuries is still largely elusive. Now, regardless of whether it's repair or regeneration, our sources confirm that all wounds, from a tiny paper cut to a major surgical incision, actually undergo the same fundamental reparative process. But there's a crucial distinction that separates the typical healing outcomes from the problematic stalled cases, isn't there?
Yes, absolutely. We differentiate between what we call acute wounds and chronic wounds. Acute wounds are those that follow an orderly and timely progression towards a sustained restoration of structure and function. They heal as expected, moving through each stage efficiently. But the real challenge in clinical practice, and where things can go significantly wrong, is with chronic wounds. These are wounds that, for various reasons, stall. They often get stuck in a prolonged inflammatory phase and simply fail to heal effectively. Understanding this distinction is absolutely critical to understanding why some wounds become long-term health burdens and require specialized interventions.
Okay, that makes sense. And to truly grasp this process, our sources break wound healing down into three distinct yet profoundly overlapping phases. It's not a strict assembly line where one phase ends completely before the next begins, right?
No, not at all. Think of it more like a meticulously choreographed dance where different parts of the orchestra play at different times, but also concurrently, creating a rich, complex harmony.
A harmony. I like that analogy.
These three core phases are inflammation, proliferation, and maturation. It helps to visualize this. Our sources use the example of a pressure sore. The initial eschar, that dark, crusty tissue that forms, reflects the immediate inflammatory phase, the body's initial damage control. As that begins to break down, you might start to see bright red, bumpy granulation tissue, which is the hallmark of the proliferative phase, where new tissue is actively being built.
Oh, okay, I can picture that.
And then the contracting or advancing edge of the wound is a key visual sign of the maturational phase, as the wound edges pull together and the scar strengthens. The key takeaway for you is that these phases are not strictly sequential, but rather overlap significantly, with their individual processes happening concurrently, sometimes even influencing each other.
All right, let's zoom in on that very first phase. Inflammation. This is the immediate response, the body's rapid reaction to injury aimed at damage control. It's a whirlwind of activity, a true biological alarm system.
It truly is. The very first thing that happens, almost instantaneously after an injury that damages blood vessels, is an intense, localized narrowing of your smallest blood vessels, your arterioles and capillaries. This vasoconstriction is your body's immediate, rapid attempt to limit blood loss. Then, quite quickly, this is followed by vasodilation, where those vessels open up, and increased vascular permeability, meaning the vessel walls become leakier.
So the vessels constrict to stop the bleeding, but then they open up and leak. That seems counterintuitive at first glance.
It does, yeah, but it's part of a precisely orchestrated sequence. That initial constriction buys crucial time. Then, specialized blood cells called platelets and your red blood cells immediately adhere to the damaged inner lining of the capillaries, the endothelium. This forms a physical plug, like a tiny dam in the capillaries, effectively stopping the hemorrhage.
How do they stick so fast?
For those who want to master the details, this platelet adhesion is primarily mediated by specific molecular hooks or glycoprotein receptors on the platelet surface, particularly one called GPI-beta, which helps them grab onto the damaged vessel. Other integrin receptors on platelets also bind to exposed collagen from the damaged vessel wall, or to other sticky proteins like fibronectin that are part of the vessel lining.
Wow. So these platelets stick, but how do they get activated to really form a solid plug and kick off this whole cascade?
Platelet activation is crucial. It happens when they bind to exposed collagen, which acts as a strong signal. This binding triggers complex internal cellular pathways within the platelets, leading to further activation and the release of powerful biologically active proteins. Platelets essentially have two main types of internal storage packets, or granules, they release. Alpha granules contain vital growth factors like platelet-derived growth factor, or PDGF, and transforming growth factor beta, or TGF-beta, along with other structural proteins. The dense bodies, on the other hand, release chemicals called vasoactive amines, like serotonin. These are primarily responsible for causing the vasodilation and increased vascular permeability we just mentioned, allowing fluid and cells to leak into the wound area.
And it's not just platelets, right? Other cells contribute to this permeability, like mast cells.
Absolutely. Mast cells, which are already present in tissues, also release histamine and serotonin, further amplifying the vessel permeability and causing plasma, the fluid component of blood, to leak from the blood vessels into the surrounding tissue. This fluid leakage is what visibly contributes to the swelling and sometimes bruising characteristic of inflammation.
Okay.
Concurrently, the coagulation cascade, your blood clotting system, is roaring to life. Activated platelets, with their unique surface properties, provide a platform for clotting factors to interact. This exponentially speeds up the production of thrombin, a powerful enzyme. Thrombin itself is a powerhouse. It activates more platelets, creating a positive feedback loop, and catalyzes the conversion of fibrinogen, a soluble protein, into fibrin, which forms long, sticky strands. These fibrin strands then trap red blood cells, forming a robust gelatinous clot that seals the wound.
The proper seal.
Exactly. And this fibrin lattice isn't just a seal, it's a crucial early scaffold for the incoming cells, like endothelial cells, inflammatory cells, and fibroblasts, essentially preparing the ground for the deeper repair work.
So it's not just about stopping bleeding. It's about immediately building a temporary foundation.
Yeah.
That's clever. And there's another cascade involved here, too, the arachidonic acid cascade.
Yes. From the breakdown of cell membranes at the injury site, substances like thromboxane A2 and prostaglandin F2 alpha are generated. These also contribute to platelet aggregation and that initial vasoconstriction. While vital for limiting blood loss, these compounds can paradoxically cause localized ischemia or reduce blood flow, potentially leading to further cell membrane damage and the release of even more of these compounds, creating a localized feedback loop that can prolong the initial damage.
Wow. Okay. Now, all this immediate, intense activity needs direction. How do cells know where to go and what to do in this chaotic environment? That's where chemokines come in, right? They're like the biological GPS system guiding the inflammatory cells.
An excellent analogy. Chemokines are small signaling proteins that act as powerful chemical attractants. They stimulate and guide the migration of different cell types, particularly inflammatory cells, into the wound site. They are incredibly active in regulating the progression through the various phases of wound healing. These chemical signals belong to specific families, and they bind to unique receptors on the surface of target cells, directing their movement.
Can you give us some crucial examples of these molecular navigators? Which ones are really key?
Certainly. One key chemokine is macrophage chemoattractant protein 1, or MCP1. As its name suggests, it's a potent attractant for monocytes, which become macrophages, and other immune cells. It's activated in skin cells after injury, and its presence is vital for bringing in the cleanup crew. Here's a crucial point for understanding chronic wounds. MCP1 expression is often sustained in them, meaning it continues to call in inflammatory cells long after they should have moved on, leading to prolonged inflammation and stalled healing.
Ah, so it gets stuck on, basically.
Exactly. Another important one is interleukin 8, or IL-8. This chemokine also helps with wound resurfacing and triggers the production of enzymes called matrix metalloproteinases, or MMPs, which are crucial for tissue remodeling. IL-8 is a strong attractant for neutrophils, driving that early inflammatory response. And here's a fascinating insight. Low levels of IL-8 are found in fetal wounds, which might be one of the reasons why fetal wounds have significantly less inflammation and heal without scars. It highlights how the balance of these signals is so important.
So some chemokines help, others can actually hinder healing if they stick around too long, or if the balance is wrong.
Precisely. For instance, CXCL10, produced by keratinocytes, is elevated in both acute wounds and chronic inflammatory conditions. This chemokine actually impairs wound healing by increasing inflammation and recruiting more lymphocytes. It also actively inhibits cell proliferation, reepithelialization, and angiogenesis, and can prevent fibroblasts from migrating where they need to be. So, while some chemokines guide repair, others, when out of balance, can actively complicate it. Finally, stromal cell-derived factor 1, or SDF1, expressed by various cells, plays a dual role. It recruits lymphocytes, contributing to inflammation, but also promotes the growth of new blood vessels and enhances the proliferation of skin cells, aiding resurfacing. You can see the incredible complexity some signals have mixed effects, depending on the context and timing.
It's all about the right signals at the right time. And the very first cells to really respond to these signals are the polymorph nuclear cells, or PMNs, more commonly known as neutrophils. These are truly the immediate first responders, rushing to the injury site.
Correct. When histamine and serotonin are released, they increase the permeability of your capillaries, literally opening pathways for these cells to squeeze through. Specific chemical factors, like certain complement proteins and leukotrienes, actively promote neutrophils to stick to the vessel walls and then engage in chemotaxis-guided movement directly to the injury site. Other inflammatory mediators, like IL-1 and TNF-alpha, further enhance the stickiness between the vessel lining and the neutrophils. Once they migrate into the inflamed area, these neutrophils start releasing the contents of their internal digestive packets, or lysosomes and enzymes, helping them move through the tissue and clear debris.
And this rapid influx of cells and fluid is what gives us those classic recognizable signs of inflammation, like redness and swelling.
Exactly. The intense vasodilation, blood vessels widening, and increased vascular permeability, vessels becoming leaky, led to those clinical findings of inflammation: rubor, redness, tumor, swelling, calor, heat, and dular pain. Local tissue swelling is further exacerbated by the deposition of fibrin, which can get trapped in the lymphatic vessels. Now, the migration of these neutrophils is an incredibly active and complex process. It requires precise, sequential sticking and unsticking interactions between molecules called integrins on the cell surface and components of the extracellular matrix.
Integrins are like those crucial molecular handholds coupled to the cell's internal skeleton, right? Allowing them to crawl almost.
Yes, that's a great way to think about them. They are crucial for cell movement, not just in wound healing and inflammation, but also in processes like embryonic development and even the spread of cancer. They allow the neutrophils, drawn by those chemokine signals, to literally crawl through the tissue by transiently gripping onto and releasing their molecular ligands or binding partners. This integrin-mediated cell motility involves four phases: adhesion, spreading, contractility or traction, and retraction. The activation of the specific integrins through binding to their targets increases cell adhesion and reorganizes the cell's internal structure, allowing it to spread out and then pull itself forward. The sophisticated regulation of integrin function provides a local control mechanism for these migrating cells.
Once these neutrophils arrive and are functionally activated, what's their primary job? I imagine it's cleaning up the mess and fighting invaders.
Their core function is indeed to scavenge for necrotic debris, dead tissue, foreign material, and, crucially, bacteria. Neutrophils have specialized receptors that allow them to recognize and engulf bacteria that have been marked for destruction by the body's immune proteins. A significant part of their bacterial killing involved generating highly reactive molecules called free oxygen radicals. They use a complex internal process to create things like superoxide anion, hydrogen peroxide, and hydroxyl radicals. While these are incredibly effective at killing bacteria, they are also a powerful double-edged sword, causing some collateral damage to the neutrophils themselves and to surrounding healthy tissue.
So a powerful but also potentially harmful tool. A bit messy. How long do these PMNs typically stay in the wound?
Their presence is temporary and relatively short-lived. Individual neutrophils survive no longer than about 24 hours and are predominantly replaced by a new wave of immune cells called monocytes. Their migration into the wound typically slows and stops after several days, or once the initial wound contamination is brought under control. However, if there's continuing wound contamination or a secondary infection, it leads to constant stimulation of the immune system, providing a steady supply of these chemotactic factors and thus a sustained influx of neutrophils. This is a significant problem. A prolonged presence of neutrophils can delay wound healing, destroy more normal tissue, and can even lead to pus formation or more serious systemic infections.
Okay, so you don't want them hanging around too long.
Definitely not. It's also important to note for you that, surprisingly, neutrophils are not absolutely essential for wound healing. Their phagocytic and antimicrobial role can actually be taken over by macrophages, which we'll discuss next. In fact, sterile incisions, those without infection, can heal perfectly normally even without the presence of neutrophils.
That leads us perfectly into the next crucial player, macrophages. If neutrophils aren't essential, but macrophages are, what makes them so special? They sound like the real stars of the show here.
The macrophage is truly considered the one truly crucial cell in wound healing. It's the orchestrator. It directs the entire show, initiating the release of critical signaling molecules, cytokines, and stimulating many subsequent processes that are absolutely vital for effective tissue repair. These tissue macrophages are derived from circulating blood cells called monocytes. These monocytes are drawn to the injury site by those same chemotactic signals we discussed, and they typically appear within 24 to 48 hours of injury, often as the neutrophils begin to disappear. Macrophages even play a role in inducing neutrophil apoptosis, essentially signaling them to die off once their immediate job is done.
So they arrive, clean up, and then take charge of the next steps. What calls them in, beyond those basic chemotactic factors?
Well, monocyte chemotactic factors are abundant and diverse, including bacterial products, C5a complement proteins, thrombin, and key proteins of the extracellular matrix like fibronectin and collagen. Important growth factors such as TGF-beta and PDGF-BB also act as strong attractants. Monocyte migration itself results from specific integrin receptors on their surface interacting with fibrin and fibronectin in the initial wound scaffold. Once activated, these integrins mediate the monocyte's transformation into a true wound macrophage, leading to increased activity in engulfing debris and pathogens, and the selective expression of various cytokines and signal transduction elements. Essentially, they become highly specialized for wound repair. Macrophages also have specific receptors for immune system proteins, allowing them to effectively identify and engulf marked pathogens.
They're clearly multitasking. What about their role beyond just gobbling up debris? Do they produce other important things?
Oh, absolutely. Activated macrophages produce nitric oxide, or NO, a molecule with diverse functions, including antimicrobial properties and effects on blood vessel dilation. When activated, macrophages induce a process that leads to the breakdown of cell membrane components, releasing other important signaling molecules. Crucially, wound macrophages release a variety of powerful proteinases, including several matrix metalloproteinases, or MMPs, such as MMP1, MEGAS2, MEGAS3, and NEGAS9. These enzymes are vital for degrading the extracellular matrix, removing foreign material and dead tissue, and promoting the movement of other cells through tissue spaces. This activity is precisely regulated and can actually be blocked by certain anti-inflammatory drugs.
This sounds like a finely tuned cleanup crew that also paves the way for new growth. But their biggest role, I understand, is in secreting those powerful cytokines and growth factors that essentially dictate the entire healing trajectory. Can you walk us through some of the most important ones?
This is truly where the macrophage shines as the orchestrator. Let's start with interleukin-1, or IL-1. This is a powerful pro-inflammatory cytokine, a key player in the body's acute response to injury or infection. It acts as an endogenous pyrogen, contributing to fever, and directly impacts blood clotting. Its effects are amplified by other signals like TNF-alpha. IL-1 enhances the production of collagen-degrading enzymes, stimulates cartilage and bone breakdown, activates neutrophils, and promotes the movement of cells. It also encourages other cells to release more pro-inflammatory cytokines, creating a chain reaction. Its influence extends into the proliferative phase, where it surprisingly boosts fibroblast and keratinocyte growth and collagen synthesis. However, here's a crucial insight. If IL-1 levels remain elevated beyond the first week, as is often seen in chronic non-healing wounds, its initially beneficial responses become maladaptive, actively hindering effective healing.
So too much of a good thing becomes bad.
Precisely. Next, tumor necrosis factor alpha, or TNF-alpha. Induced by microbial byproducts or injury, TNF-alpha is crucial for initiating the body's response. It helps immune cells stick to vessel walls and peaks around 72 hours after injury. Like IL-1, TNF-alpha contributes to fever and other inflammatory processes, and it stimulates the release of growth factors like PDGF. However, excessive or prolonged TNF-alpha production is a major problem. It's associated with multi-system organ failure and severe inflammatory states. Elevated levels are also observed in non-healing chronic ulcers. So while absolutely essential for the early inflammatory response, its persistence can severely impair wound maturation and lead to pathology.
So a common theme here seems to be good in the short term, but detrimental if it hangs around too long. What else is on the macrophage's menu of powerful secretions?
Interleukin-6, or IL-6, is produced by macrophages, monocytes, and other cells. Its increase often mirrors the neutrophil count. It acts synergistically with IL-1 and TNF-alpha, and is a potent stimulator of fibroblast proliferation vital for new tissue building. Interestingly, IL-6 levels are decreased in aging fibroblasts and in the context of fetal wounds, suggesting a role in scar development.
Interesting.
Then we have interferon gamma, or IFN gamma, another pro-inflammatory cytokine secreted by immune cells like T lymphocytes and macrophages. Its main effects include activating macrophages and neutrophils and increasing their ability to kill target cells. It also influences wound contraction and tissue remodeling. Fascinatingly, IFN gamma has been used therapeutically to treat excessive scars like hypertrophic and keloid scars, possibly by reducing collagen production and cross-linking while increasing collagen degradation. However, experimentally, high doses can impair new skin formation and wound strength. Its consistent presence as a key mediator in chronic non-healing wounds strongly suggests that T lymphocytes play a primary role in maintaining that chronic stalled state.
And macrophages are also releasing critical growth factors that really usher in the next phases of healing, correct? They sort of bridge the gap.
Absolutely. Macrophage-secreted platelet-derived growth factor, or PDGF, stimulates both collagen and other structural protein synthesis, crucial for building new tissue. PDGF-BB is the most widely studied and the only FDA-approved growth factor in this family for clinical use. Then we have transforming growth factor alpha, or TGF-alpha, and transforming growth factor beta, or TGF-beta, both released by activated monocytes. TGF-alpha stimulates epidermal growth, the resurfacing of skin and angiogenesis. TGF-beta, also released by platelets and fibroblasts, is particularly powerful. It has at least 3 major forms, or isomers: beta-1, beta-2, and beta-3. Its effects include promoting fibroblast migration and maturation and the synthesis of the extracellular matrix. TGF-beta is especially crucial for collagen metabolism and significantly accelerates healing in various experimental models. TGF-beta is known as the most potent stimulant of fibroplasia, the formation of fibrous connective tissue, and its strong cell growth effects have implicated it in the excessive fibrous tissue formation seen in diseases like scleroderma and pulmonary fibrosis.
Right, so it's linked to scarring too.
Indeed. Enhanced expression of TGF-1 is a hallmark in keloid and hypertrophic scars. Conversely, fetal wounds show a paucity or absence of TGF-beta, suggesting that scarless repair in utero might be due to low levels of this powerful fibrotic signal. Some studies even suggest that while TGF-1 and TGF-2 contribute to scarring, TGF-3 may actually limit scarring, offering a potential therapeutic avenue. As TGF-beta concentration increases in the inflammatory site, it directly stimulates fibroblasts to produce collagen and fibronectin, effectively initiating the all-important proliferative phase.
So it's not just what macrophages secrete, but the type of macrophage matters too. I've heard about M1 and M2 phenotypes. How does that play into this orchestration?
That's a crucial distinction for understanding the dynamic role of macrophages. Macrophages don't exist as a single static type. They exhibit a continuum of functional phenotypes, broadly categorized as M1, classically activated, and M2, alternatively activated. M1 macrophages are typically stimulated by bacterial components and IFN-gamma, and they release powerful pro-inflammatory mediators like TNF-alpha, nitric oxide, and IL-6. These are your frontline defenders, responsible for host defense, but they also contribute to significant tissue damage. M1 macrophages predominate in the early, highly active inflammatory phase.
Okay, the fighter.
Exactly. In contrast, M2 macrophages are activated by other cytokines like IL-4 and IL-13. They are more focused on suppressing inflammatory reactions and adaptive immune responses. They play a vital role in the later stages of wound healing, promoting angiogenesis and tissue repair. M2 macrophages predominate in the proliferative phase. The balance between these M1 and M2 phenotypes is absolutely critical for proper healing. A disturbance in this balance is widely considered a key factor in abnormal or stalled wound healing responses.
And we know macrophages are truly essential because studies have shown that their absence causes significant problems in healing. You can't just skip them.
Yes, macrophage depletion studies clearly demonstrate their importance. If you remove macrophages from the wound environment, you see delayed fibroblast infiltration into the wound, decreased wound fibrosis, and significant defects in reepithelialization, the formation of new granulation tissue, angiogenesis, and wound contraction. This shows their indispensable role in coordinating the early and mid stages of repair. Interestingly, depleting macrophages in the later stages of healing doesn't show significant differences, suggesting they are primarily required for the earlier and proliferative stages of healing, not the later maturation phase.
Finally, in this inflammatory phase, we have lymphocytes, which appear a bit later in the wound but still play an important regulatory role. What do they do?
T lymphocytes typically appear around day 5 after injury, peaking by day 7. B lymphocytes seem to be involved in downregulating healing as the wound closes. Lymphocytes can stimulate fibroblasts with cytokines, which are beneficial for new tissue growth, but they also secrete inhibitory cytokines like TGF-beta, TNF-alpha, and IFN-gamma, which can regulate or even suppress aspects of healing. Macrophages, acting as antigen-presenting cells, show bacterial debris or degraded host proteins to lymphocytes, stimulating their proliferation and further cytokine release. T cells specifically produce IFN-gamma, which in turn stimulates macrophages to release TNF-alpha and IL-1. IFN-gamma is interesting because it decreases prostaglandin synthesis, which enhances inflammatory mediators, but it also suppresses collagen synthesis and inhibits the departure of macrophages. Its consistent presence as an important mediator in chronic non-healing wounds strongly suggests that T lymphocytes are primarily involved in maintaining that chronic stalled inflammatory state. And importantly, drugs that suppress T lymphocyte functions, such as steroids and cyclosporine, impair wound healing, possibly by decreasing nitric oxide synthesis.
So once the immediate inflammatory damage control is handled, the body shifts gears into the all-important proliferative phase. This is where the actual building of new tissue begins, laying down the structural scaffolding for permanent repair.
Exactly. As the initial inflammation and blood clotting start to resolve, the wound begins to form what's known as granulation tissue. This is that characteristic red, bumpy tissue you often see filling in a healing wound. It's a dynamic, rich mixture of a brand new, developing capillary bed, meaning new blood vessels, alongside active fibroblasts, macrophages, and a loose arrangement of crucial extracellular matrix components like collagen, fibronectin, and hyaluronic acid. And this is where a host of growth factors really kick in to boost that proliferation. Things like platelet-derived growth factor, PDGF, transforming growth factor beta, TGF-beta, keratinocyte growth factor, KGF, vascular endothelial growth factor, VEGF, and epidermal growth factor, EGF, all contribute to increasing the proliferation of this new granulation tissue.
A vital part of building new tissue, especially granulation tissue, is establishing a new blood supply, right? That's angiogenesis. How does the body manage to grow new blood vessels so rapidly and efficiently? It sounds like a major construction project.
It is a major project. Angiogenesis is the process of forming new blood vessels, and it is absolutely essential to support the high metabolic demands of a healing wound. It starts with activated endothelial cells, the cells lining your existing blood vessels, degrading the basement membrane of nearby small vessels, primarily postcapillary venules. This creates gaps, allowing endothelial cells to migrate through. They then begin to divide, forming new tubules, which eventually become lumens, the hollow centers of new vessels. Eventually, a new basement membrane is deposited around these nascent vessels, leading to the maturation of these new capillaries. After injury, the endothelium is exposed to a barrage of soluble factors and comes into contact with adhering blood cells. These interactions upregulate cell surface adhesion molecules, like vascular cell surface adhesion molecule 1, making the vessels stickier. Matrix-degrading enzymes such as plasmin and various metalloproteinases are released and activated to further break down the existing endothelial basement membrane, literally clearing a path. This fragmentation allows endothelial cells to migrate into the wound, a process actively promoted by fibroblast growth factor, FGF, PDGF, and TGF-beta.
So it's a controlled breakdown to allow for rapid new construction. And then what guides the actual formation of the vessels themselves?
Precisely. Injured endothelial cells express specific adhesion molecules, like integrin ABA3, which facilitates their attachment to fibrin, fibronectin, and fibrinogen on that provisional matrix scaffold guiding their migration. Another molecule, PCAM1, on endothelial cells also modulates their interactions as they move. The actual formation of these capillary tubes is a highly complex process involving intricate cell and cell matrix interactions, again modulated by adhesion molecules on the endothelial cell surfaces. Some of these newly formed capillaries mature into arterioles and venules, while others regress through a process of involution and programmed cell death, or apoptosis, where they are subsequently removed by macrophages. The full regulation of this endothelial apoptosis surprisingly isn't completely understood.
And what about the specific growth factors and cytokines that are the primary drivers of angiogenesis? Which signals are most important here?
This process is largely stimulated and manipulated by various cytokines, predominantly produced by macrophages and platelets. TNF-alpha, released by macrophages, actually plays a significant role in orchestrating angiogenesis during the inflammatory phase itself. And a molecule like heparin can actually stimulate the migration of capillary endothelial cells by binding to and stabilizing angiogenic factors. Now, VEGF, a member of the PDGF family, is incredibly potent in promoting angiogenesis. It's produced in large amounts by various wound cells like keratinocytes, macrophages, endothelial cells, platelets, and fibroblasts. Crucially, cell disruption and hypoxia-low oxygen conditions inherent to tissue injury are strong initial inducers of powerful angiogenic factors like VEGF and its receptors at the wound site.
So low oxygen actually triggers the call for more blood vessels.
Exactly, it's a key signal. There are several VEGF family members. VEGF-A promotes early angiogenesis and is absolutely crucial for wound healing, binding to specific receptors that help organize blood vessels and drive endothelial cell proliferation. While exogenous VEGF-A can restore impaired angiogenesis, some studies show that too much can also lead to leaky, disorganized vessels, highlighting the delicate balance required. VEGF-C is also elevated in healing wounds, primarily from macrophages, and it's important in the inflammatory phase. Administering VEGF-C has shown promising results, accelerating healing in diabetic mouse models, and placental growth factor is another pro-angiogenic factor elevated after wounding, believed to work synergistically with VEGF, further enhancing new vessel growth.
So many different types of VEGF, each with its own nuance. What about FGS, the fibroblast growth factors? Do they play a role too?
Yes, they do. Both acidic and basic FGFs, FGF1 and FGF2, are released from disrupted cells and are early stimulants of angiogenesis. FGF2 provides the initial angiogenic stimulus within the first 3 days of wound repair, followed by a prolonged stimulus mediated by VEGF from day 4 through day 7. This shows a clear temporal pattern of different factors dominating at different times. Both TGF-alpha and EGF also stimulate endothelial cell proliferation. TNF-alpha is chemotactic for endothelial cells, promoting capillary tube formation, and may mediate angiogenesis by inducing a key molecule called hypoxia-inducible factor 1, HIF1, which regulates other genes involved in hypoxia response, like VEGF. There's also a positive feedback loop between endogenous nitric oxide and VEGF, with no enhancing VEGF synthesis and VEGF promoting no synthesis, so it's a tightly integrated network. TGF-beta, while a chemoattractant for fibroblasts, also assists in angiogenesis by signaling fibroblasts to produce FGFs. Other factors like angiogenic laminin, IL-8, and lactic acid also induce angiogenesis. Even components of the existing matrix like fibronectin and hyaluronic acid from the wound site are angiogenic. Ultimately, angiogenesis is a result of this incredibly complex, orchestrated interaction between ECM material and various cytokines and growth factors.
Okay, next we have fibroplasia, which is essentially the building of the structural framework of the new tissue. Where do these fibroblasts, the builders, come from? Are they circulating?
No. Fibroblasts are specialized cells that differentiate from normally quiet, resting, mesenchymal cells already present within your connective tissue. They don't migrate in from the circulation like immune cells do. After injury, these normally quiescent and sparse fibroblasts are chemically attracted to the inflammatory site. They then begin to divide rapidly and produce the essential components of the extracellular matrix. Stimulated by macrophage-derived and platelet-derived cytokines and growth factors, for example, platelet-derived TGF-beta indirectly stimulates fibroblast proliferation by releasing PDGF. The fibroblast, normally in a resting state, enters active replication and proliferation. Fibroblasts can even stimulate their own replication in a positive feedback loop by releasing FGF2. For continued high-level proliferation, they typically need further stimulation from factors like EGF or IGF-1, though they can survive at a lower level without these growth factors.
So their main job is to crank out collagen, the ultimate structural protein. That's their primary function.
Exactly. Their primary function is to synthesize collagen, and they actually begin this process during the cellular phase of inflammation, surprisingly early. There's a crucial delay, generally 3 to 5 days, between the injury and the first significant appearance of new collagen in the healing wound. This is called the lag phase of wound healing, and it's the time required for those undifferentiated mesenchymal cells to fully transform into highly specialized active fibroblasts. Fibroblasts migrate in response to a wide array of chemotactic substances, including various growth factors like PDGF and TGF-beta, components of the immune system, clotting factors like thrombin, and even fragments of collagen and fibronectin itself.
And this collagen production eventually winds down and matures, making the scar stronger over time.
Yes. The rate of collagen synthesis in the wound starts to decline after about 4 weeks, and eventually balances with the rate of collagen destruction by enzymes like collagenase or MMP1. At this point, the wound enters a phase of collagen maturation and remodeling, which is a slow process that can continue for months or even years. During this maturation phase, other components of the extracellular matrix, like glycoproteins and mucopolysaccharides, decrease, and that dense network of new capillaries that formed in the proliferative phase regresses and disappears. These changes collectively alter the appearance of the wound and, critically, significantly increase its strength over time.
Finally, in this proliferative phase, we have epithelialization, which is the crucial process of resurfacing the wound, creating that protective barrier again that separates us from the outside world. This seems absolutely vital.
It is fundamental for protection. Your epidermis acts as a vital physical barrier, preventing fluid loss from inside and bacterial invasion from outside. Tight cell junctions within the epithelium contribute to its impermeability. The basement membrane zone, or BMZ, is also crucial here. It provides structural support and anchors the epidermis to the underlying dermis. The BMZ itself is made of several layers of specialized proteins, including laminin, heparin sulfate, and various types of collagen, all arranged to provide strong attachment.
How quickly does this resurfacing begin after an injury? Is it immediate?
It starts remarkably quickly, often within hours after injury. The wound is first rapidly sealed by the initial blood clot, and then by the active migration of epithelial cells, specifically keratinocytes, across the defect. These keratinocytes, which are the main cells of the epidermis, migrate from the basal layer of the remaining healthy epidermis, or from the depths of epithelium-lined structures, like hair follicles within the dermis. This process involves a sequence of changes in these wound keratinocytes. They detach from their neighbors, migrate across the wound bed, followed by rapid proliferation, multiplying, differentiation, maturing into specialized skin cells, and finally stratification, forming new epidermal layers. If the basement membrane zone is still relatively intact, epithelialization proceeds much more rapidly because it provides a smooth, guided path.
So they essentially unstick themselves and then start moving across the gap.
Yes, that's a good way to put it. Epidermal cells express specialized integrin receptors, allowing them to interact dynamically with extracellular matrix proteins like fibronectin, which helps guide their movement. These migrating cells literally dissect the wound, separating the desiccated eschar, or scab, from the viable, healthy tissue underneath. The specific path they take is influenced by the integrins they express and the signals they encounter. To move efficiently, they also need to degrade some of the existing extracellular matrix in their path, which they do by producing collagenase and MMP1 and other enzymes that help clear the way. As they migrate, these cells are also somewhat phagocytic, meaning they actively remove small debris. Once the leading edges of cells from opposite sides meet, the cells behind them begin to rapidly proliferate, filling in the new layers. This movement is often described as a leapfrog and tumbling fashion.
Leapfrog and tumbling, huh?
Yeah. If the basement membrane zone wasn't intact, the body will prioritize repairing it first as the cells move over the wound bed. The absence of neighboring cells at the wound margin may be a key signal for epidermal cell migration and proliferation. Local release of growth factors like epidermal growth factor, EGF, TGF-alpha, and keratinocyte growth factor, KGF, along with increased expression of the receptors, also actively stimulate these processes. Topical application of KGF2 has even been shown to accelerate reepithelialization in animal models. Once the wound is completely reepithelialized, basement membrane proteins like laminin reappear in a highly ordered sequence. The cells then become columnar, stratified, and firmly reattached to the newly reestablished BMZ and the underlying dermis.
That's a truly detailed picture of the phases, but none of this complex cellular choreography happens in isolation. It all relies critically on the extracellular matrix, or ECM. This isn't just a passive scaffold, is it? Our sources suggest it's much more dynamic than that.
No, not at all. The ECM is incredibly dynamic and active. While it certainly provides the physical structure and stability for tissues, it also plays an active and complex role by profoundly regulating the behavior of the cells that interact with it. The cells within the ECM are the ones producing its macromolecular constituents. These primarily include glycosaminoglycans, or Js, which are long polysaccharide chains often linked to proteins as proteoglycans, and a variety of fibrous proteins like collagen, elastin, fibronectin, and laminin.
So the cells are building their own environment, and that environment, in turn, is telling them what to do and how to behave. What makes up this ground substance that gives the tissue its specific properties, like its sort of gel-like feel?
Right. In connective tissue, those proteoglycan molecules form a gel-like ground substance. This gel is highly hydrated, primarily because of the numerous negatively charged groups on the GAGs, which strongly attract positively charged ions like sodium, which in turn draws in large amounts of water. This remarkable hydration is what gives the matrix its turgor, allowing it to withstand compressive forces without collapsing. It also permits the rapid diffusion of vital nutrients, metabolites, and hormones between your blood and tissue cells. Within this gel, collagen fibers are meticulously organized to strengthen the matrix, while elastin fibers provide its crucial resilience and ability to snap back into shape.
And the wound matrix isn't static. It constantly evolves during healing, right? It changes composition.
Exactly. The wound matrix continually accumulates new components and changes in composition over time in a constant delicate balance between new deposition and enzymatic degradation. The provisional matrix is the initial temporary scaffold laid down immediately after injury. It's composed of fibrin, fibrinogen, fibronectin, and vitronectin, providing a sticky base for cells to migrate on. GAGs and proteoglycans are synthesized next, providing vital support for further matrix deposition and remodeling. The end result, which are the predominant structural proteins of the final scar, are collagens. Other crucial attachment proteins like fibrin and fibronectin provide crucial linkages to the ECM by binding to specific molecular hooks on cell surfaces called integrin receptors. The stimulation of fibroblasts by growth factors, for instance, actually increases the expression of these integrin receptors, facilitating stronger cell matrix interactions. Ligand binding then causes these integrins to cluster into concentrated focal adhesion sites, which are key points of cell attachment. Even extracellular divalinications like magnesium and calcium regulate integrin function by inducing conformational changes, essentially activating or deactivating their binding ability.
This sounds like a constant, dynamic conversation between the cells and their environment, influencing each other back and forth.
It is a profound and dynamic reciprocal relationship. For instance, the way fibroblasts respond to cytokines, those cell signaling molecules, is actually altered by variations in the composition of the ECM itself. This is a feedback loop. For example, the expression of matrix-degrading enzymes like the MMPs is significantly upregulated after cytokine stimulation of fibroblasts. Collagenolytic MMP1 specifically is induced by IL-1, but surprisingly downregulated by TGF-beta. The activation of other enzymes that break down the matrix is also critical. All of this controlled degradation is essential because it literally carves a path, facilitating cell migration into and through the wound. This dynamic modulation of ECM components provides additional, very precise local control mechanisms over the entire healing process. We even see this matrix modulation go awry in disease states like tumor metastasis, where cancer cells lose their normal attachment to the ECM, which is mediated by integrins and fibronectin, allowing them to break away and spread throughout the body.
Can you give an example of this dynamic interaction in action, perhaps a clinical one, to really bring it to life, something we might see used?
Yeah, a great example is how TGF beta influences healing in incisional wounds treated with fibrin sealant. Fibrin sealant is a plasma derivative that mimics the final step in the blood clotting cascade, but it's much more concentrated, creating an airtight, waterproof seal. While it's beneficial for sealing, it can also act as a mechanical barrier, blocking early cell-mediated wound healing. However, when fibrin sealant is supplemented with TGF-beta, studies have shown it can actually reverse these inhibitory effects and even increase the tensile strength of the wound compared to simply suturing alone.
Really? So adding the growth factor helps overcome the sealant's blocking effect.
Exactly. This increased strength is thought to result from improved cell migration into the wound, faster clearance of the fibrin sealant itself, suppression of a specific MMP, MMP9, and enhanced ECM synthesis in those TGF-beta supplemented wounds. It beautifully illustrates how simply adding one signaling molecule can dramatically change the dynamic.
That's a perfect illustration. Now let's talk about the ultimate structural backbone of the ECM, collagen. It's everywhere in our body, right? It's incredibly abundant.
Absolutely. Collagen is arguably the most abundant protein in the human body, constituting a massive 25% of the total protein mass in mammals, being a major component of skin, bone, tendons, and cartilage. Structurally, the collagen molecule is a long, stiff, triple-stranded helical structure composed of three intertwined polypeptide chains called alpha chains, wind around each other like a rope. Specific amino acids like proline and glycine are crucial for the stability and tight packing of this unique structure. There are at least 20 known types of collagen, each with slightly different structures and functions. Types 1, 2, 3V, and A other are the main constituents of connective tissue. Type I is the principal collagen of skin and bone and is the most common, making up about 80% of adult skin. Type 3, on the other hand, makes up about 20% of adult skin, but is found in higher concentrations in newborns and is also highly expressed in early wound healing, contributing to the initial flexibility of new tissue. Fibrillar collagens, like type 1, are secreted into the extracellular space where they self-assemble into thin collagen fibrils, which then aggregate into larger cable-like bundles called collagen fibers.
So different types for different functions and different stages of life or healing.
Yes. Other types include fibril-associated collagens, like types 9 and 12, found on the surface of collagen fibrils, linking them to each other and to other ECM components. Network-forming collagens, types 4 and 7, are also critical. Type 4 molecules assemble into a mesh-like pattern, forming a major part of the basal lamina, that specialized foundation we discussed. Type 7 forms anchoring fibrils that help attach the basal lamina to the underlying connective tissue, especially abundant in the skin. There are also collagen-like proteins, such as type 7, found in crucial cell adhesion structures, and type 8, located in the basal laminae of blood vessels. Interestingly, a powerful anti-angiogenic peptide called endostatin, which inhibits the growth of new blood vessels and shows promise as an anti-cancer drug, is formed by cleavage of a part of type 8 collagen.
How does the body actually synthesize all this collagen? It sounds incredibly intricate, like building ropes inside your cells.
Collagen synthesis is indeed a highly complex multi-step process. The individual collagen polypeptide chains, called pro-alpha chains, are first synthesized inside the cell. They then enter the endoplasmic reticulum, where a crucial step occurs. Certain proline and lysine amino acids undergo hydroxylation, the addition of hydroxyl groups. This hydroxylation is absolutely critical because it enables the formation of the stable triple-stranded helix through interchain hydrogen bonds, giving collagen its strength. After this, three pro-alpha chains combine to form procollagen, which is the full triple-stranded helical molecule. A crucial side note here. In conditions like vitamin C deficiency or scurvy, proline hydroxylation is prevented. This leads to the formation of unstable triple helices because the collagen structure is defective, resulting in fragile blood vessels, loose teeth, and impaired wound healing, as existing normal collagen gradually degrades and cannot be properly replaced.
So vitamin C is absolutely essential for collagen stability and thus for healing. That's a key takeaway.
Absolutely. After secretion from the cell into the ECM, specific enzymes called proteases cleave off parts of the collagen molecules, forming mature collagen monomers. These monomers then spontaneously self-assemble into collagen fibrils in the ECM, a process driven by collagen's natural tendency to organize. Finally, covalent cross-linking of the lysine residues between collagen molecules provides the immense tensile strength that collagen is known for. The extent and type of cross-linking vary by tissue. In tendons, for example, where extremely high tensile strength is critical, cross-linking is very extensive. In mammalian skin, collagen fibrils are organized in a basket weave pattern to resist multidirectional stress, while in tendons, they're in parallel bundles aligned with the major axis of tension.
What other factors can affect collagen synthesis beyond vitamin C? Are there things that boost it or slow it down?
Besides vitamin C, growth factors like TGF-beta, IGF-1, and IGF-2 all increase collagen synthesis. Conversely, IFN-gamma decreases the production of type I procollagen, and powerful drugs like glucocorticoids inhibit the basic gene transcription for procollagen, leading to significantly decreased collagen synthesis. There are also genetic disorders directly linked to collagen formation. Osteogenesis imperfecta, for instance, often called brittle bone disease, is caused by a genetic defect that leads to weak, easily fractured bones due to faulty collagen. Ehlers-Danlos syndrome results from mutations affecting type 3 collagen, characterized by fragile skin, fragile blood vessels, and hypermobile joints. These conditions highlight the critical role collagen plays in overall body integrity.
From the structural backbone in collagen, let's move to the components that give tissues their springiness. Elastic fibers, what's their main role, particularly in skin? Why are they important?
Elastic fibers are crucial for tissues like skin, blood vessels, and lungs that require both strength and the ability to stretch and recoil. They provide the resilience needed for tissues to snap back into shape after temporary stretching. These fibers are predominantly composed of elastin, a highly hydrophobic protein. Soluble forms of elastin are secreted into the extracellular space, where they spontaneously form crucial crosslinks with other elastin molecules, creating a large, interconnected network of elastin fibers and sheets. Elastin's unique structure, with alternating flexible and more rigid segments, is responsible for its remarkable elastic properties. Think of it like a naturally occurring rubber band in your tissues. Elastic fibers consist of an elastin core covered by a sheath of very fine structures called microfibrils, which are composed of glycoproteins like fibrillin. Fibrillin is absolutely essential for the integrity and proper formation of these elastic fibers.
So the microfibrils are like the essential scaffolding or framework for the elastin to be laid upon. They set the pattern.
Exactly. Microfibrils actually appear before elastin in developing tissues, seeming to form a scaffold onto which elastin is precisely deposited. Elastin is produced early in life, then stabilizes, and doesn't undergo much further synthesis or degradation through adulthood, with a very slow turnover rate. Age-related modification results from progressive degradation, as elastic fibers gradually become tortuous, frayed, and porous, contributing to the loss of skin elasticity as we age. Interestingly, in normal, unwounded circumstances, there's very little elastin degradation, likely due to its hydrophobic nature, which makes its interior largely inaccessible to enzymes. Because of its complex three-dimensional structure and extensive cross-linking, significant enzymatic cleavage is required to lose much elasticity. Growth factors like IGF-1 and TGF-beta stimulate elastin production, while glucocorticoids and basic FGF can reduce it in adult skin cells.
And like collagen, there are genetic disorders tied to elastin, correct?
Right.
But things go wrong genetically.
Yes. Mutations causing an elastin protein deficiency can lead to significant arterial narrowing due to excessive smooth muscle cell proliferation in the arterial wall. This suggests that the normal elasticity of an artery is necessary to prevent this uncontrolled cell growth. Also, gene mutations in fibrillin lead to Marfan syndrome, a serious connective tissue disorder that makes affected individuals prone to aortic rupture and other cardiovascular issues due to defective elastic fibers. These conditions really underscore the vital role of elastin in tissue integrity.
So far, we've covered the fibers, proteins, collagen, and elastin. Now let's explore glycosaminoglycans, GAGs, and proteoglycans, which give the matrix its gel-like properties and its incredible ability to hydrate and absorb shock.
Right. GAGs are unbranched polysaccharide chains composed of repeating sugar units, and they're highly negatively charged due to sulfate or carboxyl groups. There are several main types, including hyaluronan, chondroitin sulfate, and heparin sulfate. In connective tissue, GAGs make up less than 10% of the weight of fibrous proteins, but their high negative charge is incredibly important. It strongly attracts positively charged ions like sodium, which in turn draws massive amounts of water into the matrix. This creates a porous, highly hydrated gel that's responsible for the turgor, the firmness or plumpness of tissues, allowing the matrix to withstand compressive forces without collapsing. This gel also permits the rapid diffusion of vital nutrients, metabolites, and hormones between your blood and tissue cells.
And hyaluronan is especially important, isn't it? Particularly in the context of fetal healing. You mentioned that earlier.
It is. Hyaluronan is the simplest JAG. It's not sulfated, and while found in adult tissues, it's particularly prevalent in fetal tissues. Its abundance in fetal wounds is considered a key factor in the scarless wound healing observed in utero. Unlike other JAGs, hyaluronan isn't covalently attached to any protein. It's synthesized directly on the cell surface. It plays several crucial roles due to its large hydration shell. It's produced in large quantities during wound healing, facilitating cell migration by literally physically expanding the ECM and providing space, and also by reducing the adhesion strength of migrating cells to the matrix fibers. In embryogenesis, hyaluronan can create cell-free space to allow for cell migration. Once cell migration is complete, excess hyaluronan is degraded by enzymes called hyaluronidases. Therapeutically, hyaluronic acid derivatives have shown promise in accelerating healing in burns, surgical wounds, and chronic wounds.
What about proteoglycans themselves? How do they function beyond just holding water?
Proteoglycans are a diverse group of glycoproteins, proteins with attached sugar chains, whose functions are mediated by their central core proteins and those attached GAG chains. They vary greatly in the number and types of GAGs attached, and these GAGs can be further modified, for example, by sulfonation, which changes their charge and function. Because of their GAGs, proteoglycans provide critical hydrated space around and between cells, forming gels with varying pore sizes and charge densities to regulate cell and molecule movement. For example, perlecan, a heparin sulfate proteoglycan, plays this role in the basal lamina of the kidney glomerulus, acting as a molecular filter. Decreased levels are implicated in diabetic albuminuria.
So they do more than structure, they're involved in signaling too.
Yes, critically. Beyond structural roles, proteoglycans are crucial in chemical signaling. They can bind various secreted signal molecules, like growth factors, profoundly modulating their signaling activity. They can also bind other secreted proteins, such as proteases and protease inhibitors. This binding allows proteoglycans to regulate these proteins by immobilizing them, providing a reservoir for delayed release, altering the protein for better receptor presentation to a cell, prolonging its action by protecting it from degradation, or even blocking its activity entirely. Some proteoglycans are even components of cell membranes, acting as co-receptors with other cell surface receptor proteins to help cells bind to the ECM and initiate specific cellular responses to extracellular signals. For instance, syndicins are transmembrane proteoglycans found on fibroblasts and epithelial cells. In fibroblasts, they interact with fibronectin on the cell surface and with the cell's internal cytoskeleton and signaling proteins, demonstrating a direct link between the outside and inside of the cell. Mutations in these co-receptor proteoglycans can result in severe developmental defects, highlighting their fundamental importance.
It's clear the ECM is a very busy, dynamic place, packed with interacting molecules. What about other non-collagen proteins like fibronectins? What do they do?
Fibronectins are another critically important class of non-collagen proteins in the ECM. They have multiple distinct regions or domains that allow them to bind to other matrix macromolecules and to cell surface receptors. These interactions are fundamental for organizing the matrix and facilitating cell attachment and migration. Fibronectin exists in two main forms: soluble plasma fibronectin, which circulates in body fluids and enhances blood clotting, wound healing, and the immune system's ability to engulf foreign particles, and insoluble fibrillar forms, which assemble on cell surfaces and are then deposited into the ECM, forming the stable tissue structure. The fibronectin fibrils that form on fibroblasts are usually physically coupled with the cell's internal actin stress fibers, which are part of the cell's internal skeleton. These actin filaments actually promote the assembly of fibronectin fibrils and influence their orientation, showing that the cell actively shapes its environment. Crucially, integrin transmembrane adhesion proteins mediate these interactions. The contractile actin and myosin cytoskeleton within the cell then pulls on the fibronectin matrix, generating tension within the tissue, which is vital for processes like wound contraction.
Then there's the basal lamina, that specialized foundation beneath epithelia. What is its unique structure and critical function? Sounds like a really important boundary layer.
It is. The basal laminae are flexible, remarkably thin mats of specialized ECM, typically separating cells and epithelial layers from the underlying or surrounding connective tissue. In the skin, for example, the basal lamina is firmly tethered to the underlying connective tissue by specialized anchoring fibrils. This composite of basal lamina and deeper collagen is often referred to as the basement membrane. While its precise composition can vary by tissue, most mature basal laminae contain type 4 collagen, a unique, flexible mesh-forming collagen, along with the large proteoglycan prolican and crucial glycoproteins called laminin and nitogen. Type 4 collagen has a more flexible structure than the fibrillar collagens, with its triple-stranded helix interrupted by bends, allowing it to form a flexible mesh. Laminin generally consists of 3 long, intertwined polypeptide chains. The laminin gamma-1 chain is particularly crucial for basal lamina formation, as mice lacking it cannot form a basal lamina and die during embryogenesis. Laminin in basement membranes has specific regions that bind to prolican, nitogen, and various cell surface laminin receptors. The type IV collagen and laminin networks are further connected by a nitogen and prolican, acting as stabilizing bridges that link these key components together. Many cell surface receptors for type IV collagen and laminin are integrins, further highlighting the importance of these cell ECM interactions. Another important laminin receptor is dystroglycan, a transmembrane protein that, along with integrins, helps organize basal lamina assembly.
So it's not just a physical separator between tissue layers. It has other jobs too.
Not at all. It serves numerous vital functions. First, it acts as a molecular filter, precisely preventing the passage of macromolecules, as seen famously in the kidney glomerulus, which filters blood. Second, it's a selective barrier to certain cells. For example, the basal lamina beneath the epithelium typically prevents fibroblasts from contacting epithelial cells, but it does allow immune cells like macrophages or lymphocytes to cross when needed. Third, and critically for wound healing, it serves as a crucial scaffold for regenerating cells to migrate along, guiding their path of movement. And importantly, it's a critical element in tissue regeneration in locations where the basal lamina survives the injury, acting as a template for new tissue to perfectly regrow.
So much construction, building up this complex matrix, but also there has to be a precise mechanism for breaking down the ECM. Degradation is just as important as building, right? For processes like cell migration, you need to clear a path.
Absolutely. The highly regulated turnover of the ECM is crucial for a vast array of biological processes, not just healing. ECM degradation occurs, for instance, during metastasis, when cancer cells migrate from their origin to distant organs. In the context of injury or infection, localized degradation of the ECM allows cells to migrate across the basal lamina to reach the site of the problem. This degradation is carried out by locally secreted cellular proteases, such as the MMPs or serine proteases, which break down specific ECM components. This matrix proteolysis helps the cell migrate by literally clearing a path through the existing matrix, by exposing new binding sites that promote cell binding or migration, by facilitating cell detachment so a cell can move forward, and by releasing powerful signal proteins that promote cell migration.
And this breakdown is tightly regulated. It can't just be a free-for-all, otherwise you'd have rampant tissue destruction, surely.
No, it's under very, very tight control, which is essential to prevent widespread tissue damage. Many proteases are secreted as inactive precursors and are only activated when and where they're specifically needed. Additionally, cell surface receptors bind these proteases, ensuring their action is highly localized to the precise sites where they're required, preventing them from floating around and causing unintended damage. Finally, there are specific protease inhibitors, such as the tissue inhibitors of metalloproteinase, or TIMPs, which can bind these enzymes and block their activity. This multi-layered control ensures that degradation is purposeful and precise.
Once the body has meticulously built new tissue during the proliferative phase, the final and often longest phase is maturation. This is where the scar really develops, reorganizes, and strengthens. And here we need to understand a critical distinction between wound contraction and wound contracture. They sound similar, but one is good, the other's a problem.
This is a critical distinction, both biologically and clinically. Wound contraction is a natural, beneficial process. It's the centripetal movement, meaning pulling inward from the edges of the surrounding full-thickness skin, working to reduce the overall amount of disorganized scar tissue that needs to be formed. It's your body's way of minimizing the size of the defect that needs to be filled, making the wound smaller.
Okay, so the body tries to shrink the gap.
Exactly. In contrast, wound contracture is an adverse and problematic outcome. It's a physical constriction or limitation of function resulting from excessive scar tissue that goes beyond normal wound contraction. This leads to functional disability, such as scars that cross joints and prevent full extension, or scars involving the eyelid or mouth that pull the tissue away, causing ectropion. It's when the healing process, intended to be beneficial, becomes dysfunctional and problematic, impeding normal movement or appearance.
So how does this wound contraction, the beneficial kind, actually happen? What are the mechanisms at the cellular level? It sounds like the tissue is actively pulling itself together.
It is. The precise mechanisms are still an active area of research and not completely understood, but it's a complex interaction between the cells and the extracellular matrix. Experiments using fibroblast-populated collagen lattices in the lab have shown that fibroblasts, in their attempt to move and attach, become trapped by collagen fibers, causing the fibers to bunch and contract. This suggests that the pulling action of these cells plays a key role. Crucially, fibroblasts within a contracting wound undergo a remarkable transformation into specialized cells called myofibroblasts. These cells share characteristics and functions with both regular fibroblasts and smooth muscle cells, and they express alpha smooth muscle actin within their internal structures, organized into bundles called stress fibers. This actin typically appears around day 6 post-wounding, remains at high levels for about 15 days, and then typically disappears by 4 weeks, often through programmed cell death or apoptosis.
So the fibroblasts actually develop a muscle-like ability to pull the wound edges together, literally shrinking the wound.
That's amazing. Precisely. A stimulated fibroblast develops this contractile ability, which is directly linked to the formation of internal cytoplasmic actin-myosin complexes, similar to what makes muscle cells contract. When these stimulated cells are placed in a laboratory collagen lattice, the contraction occurs even more quickly, directly demonstrating their pulling power. The tension exerted by the fibroblasts attempting to contract seems to stimulate these actin-myosin structures within their cytoplasm. In laboratory settings, if you add drugs that inhibit the cell's internal skeleton, like colchicine, which affects microtubules, or cytocholisin D, which affects microfilaments, you see minimal contraction of the collagen gels, highlighting the essential role of the cytoskeleton in this process. These fibroblasts also tend to arrange themselves linearly along the lines of tension in the wound, and if that tension is removed, the cells tend to revert to a more rounded shape.
And these myofibroblasts show up in diseases too, not just normal healing.
Yes, they are abundantly present in diseases involving excessive fibrosis in many organs, such as hepatic cirrhosis, renal and pulmonary fibrosis, Dupuytren contracture in the hand, and in the dense fibrous reactions seen around cancers. The actin microfilaments within them are arranged linearly along the long axis of the fibroblast and are associated with dense bodies that allow attachment to the surrounding ECM, forming a direct link between the cell's internal machinery and its external environment. This attachment entity, connecting the cell's cytoskeleton to the ECM and spanning the cell membrane, is sometimes called the fiber nexus.
Do MMPs, those matrix-degrading enzymes, also play a role in contraction, or is it all about the pulling?
No, MMPs are important here too. Stromolysin 1, or MMP3, has been shown to strongly affect wound contraction. It's thought that MMPs may be necessary to precisely cleave the attachments between the myofibroblasts and the collagen fibers, allowing the collagen lattice to contract more efficiently. Different fibroblast populations from different organs can respond to this contractile stimulus in varied ways. It's likely that stromolysin 1, working in concert with integrins on the cell surface, modifies the attachment sites between fibroblasts and collagen fibrils, facilitating the pulling. Similarly, cytokines like TGF-1 affect contraction by increasing the expression of crucial integrins.
After all that contraction, the final long stage is the actual remodeling of the scar. What happens during this phase, which can go on for months or even years? How does the scar change?
During remodeling, the fibroblast population gradually decreases in number, and that dense capillary network that formed in the proliferative phase also regresses, making the scar less red. The wound strength increases rapidly within the first 1 to 6 weeks, then appears to plateau, continuing to improve slowly for up to a year or even longer. However, a crucial point for you to understand is that even a fully healed scar only achieves about 30%, maybe 80% at best, but often cited around 30-50% of the tensile strength of non-wounded original skin.
Only 30%. That's surprisingly low.
It is. A major increase in the scar's breaking strength occurs after approximately 21 days, primarily due to the ongoing collagen cross-linking, the formation of strong chemical bonds between collagen molecules. While this cross-linking significantly increases strength and can cause further wound contraction, it also results in a scar that is more brittle and less elastic than normal skin, making it less flexible. Another significant difference is at the interface between the epidermis and dermis. A healed wound lacks reap pegs, those undulating epidermal projections that penetrate into the papillary dermis of healthy skin. This loss of this complex interdigitating anchorage makes the new epidermis more fragile and predisposes it to avulsion, or tearing away after even minor trauma. So the scar is functional but fundamentally different from original tissue.
Given how incredibly complex all these interwoven events are, it's not surprising that abnormal wound healing is a significant and common challenge. So many factors can impede the desired outcome, can't they?
Indeed. The final scar type, whether it heals adequately, inadequately, or becomes proliferative, is dictated by a delicate and dynamic balance between new collagen deposition and existing collagen degradation. If that balance is tipped in either direction, too much production or too little degradation or vice versa, the result is often poor. General factors that inhibit healing include the sheer amount of tissue lost or damaged, the presence of foreign material or bacterial infection, and the duration of exposure to any toxic factors in the wound environment. Intrinsic factors within the patient also play a huge role, such as age, the use of certain chemotherapeutic agents, underlying medical conditions like atherosclerosis, heart or kidney failure, and even the wound's specific location on the body.
The most common forms of abnormal healing that people are probably familiar with are hypertrophic scars and keloids. What exactly are these, and how do they differ because they often get confused?
That's a common point of confusion. Both hypertrophic scars and keloids are classified as proliferative scars, meaning they result from an excessive net deposition of collagen. However, they have distinct characteristics that are important to differentiate. Hypertrophic scars are raised scars that remain strictly within the confines of the original wound. They often appear red and itchy, but crucially, they show a tendency to regress spontaneously over time, meaning they can fade or flatten on their own. Keloids, on the other hand, are much more problematic and severe. By definition, they grow beyond the borders of the original wound, invading surrounding healthy tissue, and they rarely regress spontaneously. They are significantly more prevalent in darkly pigmented skin, developing in 15% to 20% of African Americans, Asians, and Hispanics, and there's strong evidence of genetic susceptibility, including familial inheritance and common occurrence in identical twins. Recent studies even suggest a strong multigenetic disposition, meaning multiple genes likely contribute.
So a strong genetic component, especially for keloids. That makes them much harder to treat, I imagine. What biological pathways are being investigated to understand keloids better?
Researchers are actively looking at proposed pathways involving programmed cell death, apoptosis, how cells engulf material, endocytosis, the intricate interactions between cytokines and their receptors, specific cell signaling pathways like the MAPK pathway, as well as the roles of proteins like tenascin C, specific proto-oncogenes, and various growth factors like TGF-beta and VEGF. Keloids often appear in specific high-tension locations on the body, such as above the clavicles, on the trunk, upper extremities, and face. Their occurrence is unpredictable, and they are frequently resistant to both medical and surgical intervention, with no known single effective prevention method currently. However, recent research has identified potential molecular and genetic targets for prevention, such as specific transcriptional factors that control gene expression.
Are there clear differences under the microscope between a normal scar, a hypertrophic scar, and a keloid? Can you tell them apart visually that way?
Yes, there are distinct histological differences. In a normal scar, collagen bundles are randomly arrayed and appear relaxed. Hypertrophic scars primarily contain well-organized type 3 collagen, with islands of fibroblasts, small vessels, and collagen fibers distributed throughout the dermis. Keloids, in contrast, feature disorganized bundles of both type I and type 3 collagen. They have much thicker, abundant collagen bundles that often form acellular, nodule-like structures deep in the dermis, with a surprising scarcity of cells centrally within those nodules. Both hypertrophic scars and keloids, however, tend to have stretched collagen bundles aligned in the same plane as the epidermis, reflecting the underlying tension.
So hypertrophic scars can often be prevented or are reversible. What causes them, if not primarily genetics like keloids?
Hypertrophic scars are indeed often preventable. Factors like prolonged inflammation and insufficient resurfacing, which are common in severe burn wounds, actively promote their formation. A significant and often overlooked cause is tension. Scars oriented perpendicular to underlying muscle fibers tend to be flatter and narrower, with less collagen formation compared to those placed parallel to the muscle fibers. Tension appears to act as a powerful signal, activating fibroblasts and leading to excessive collagen deposition.
Ah, so the direction of the scar matters a lot.
It does. This highlights why the position of an elective surgical scar can be chosen carefully to induce a narrower, less obvious healed scar. As muscle fibers contract, wounds placed perpendicular to them are brought closer together, while those parallel tend to pull apart, leading to greater tension and more scar formation. Hypertrophic scars represent a reversible hyperproliferative phenotype that tends to regress if the original stimuli, like chronic skin tension or an excess of stimulatory growth factors, are removed.
But keloids are a different beast then, more stubborn, genetically driven.
They are. Keloids appear to be genetically predisposed, and once activated, they seem to be irreversibly switched on by factors like TGF-beta. In both hypertrophic scars and keloids, collagen synthesis is elevated, meaning too much collagen is being made, and critically, collagen degradation is low, meaning it's not being broken down sufficiently. The balance is off. Specifically, MMP1, a collagen-degrading enzyme, and MMP9, another enzyme important in early tissue repair, are decreased, while MMP2, an enzyme important in late tissue remodeling, is significantly elevated. This imbalance contributes to the excessive collagen accumulation. Blocking TGF-beta activity with antibodies has been shown experimentally to decrease scar fibrosis. Research also suggests that increased mechanical stress at the wound bed actively promotes hypertrophic scarring by activating mechanotransduction pathways, the ways cells sense and respond to physical forces. So targeting these pathways may be a future strategy to reduce excessive scarring.
What are the current practical prevention strategies for both types of proliferative scars? What can people do right after an injury or surgery?
The three main strategies immediately after wound closure are tension relief, hydration and occlusion, and the use of taping or pressure garments. For wounds with higher tension or in high-risk areas like the deltoid and sternal regions, or those perpendicular to lines of skin tension, post-surgical taping for up to 3 months can significantly reduce scarring by reducing tension. Moisturizing lotions and moisture-retentive dressings, especially silicone sheets and gels, are highly effective. They can reduce scar thickness, discomfort, and itching, and significantly improve appearance. Scar tissue loses water more rapidly than normal skin, and silicone products can prevent these evaporative losses, aiding hydration and creating a favorable healing environment. These should be applied as soon as the initial wound has closed. Pressure garments are used prophylactically, especially for wide wounds like severe burns that take more than 2-3 weeks to heal, and should be applied as soon as the wound is closed. The mechanism is believed to involve reducing oxygen tension in the wound, which decreases myofibroblast proliferation and collagen synthesis. Pressure therapy likely acts on cellular mechanoreceptors, influencing fibroblast apoptosis. Lastly, avoiding sun exposure and using SPF 50+ sunscreen for at least a year postoperatively is crucial to reduce scar hyperpigmentation and improve cosmetic outcome.
And for existing linear hypertrophic scars, what's the typical treatment path if prevention didn't work or wasn't possible?
For early linear scar hypertrophy, typically from 6 weeks to 3 months after injury, pressure therapy is often recommended. After 6 months, silicone therapy should be continued. If hypertrophy persists and is problematic, intralesional corticosteroids, specifically triamcinolone acetonide, injected directly into the scar tissue every 2 to 4 weeks, is the only non-invasive management option with strong evidence of effectiveness. It has a high response rate, but recurrence can be up to 50%.
Oof, 50% recurrence is high.
It is. Adverse effects include skin thinning, hypopigmentation, loss of color, tiny visible blood vessels, and pain, so injections must be precisely limited to the scar itself. For permanent linear hypertrophic scars after 1 year, surgical scar revision might be considered, often combined with techniques like undermining adjacent tissue, carefully placed deep sutures, or Z-plasty, which rearranges the scar to release tension. This is always followed by continued post-surgical taping and silicone therapy to prevent recurrence. Scar contractures, which cause functional impairment, particularly across joints, generally require surgical correction, often with Z-plasty, skin grafts, or flaps, to release the tension and restore function.
What about widespread hypertrophic scars, like those from severe burns? Those must be incredibly challenging to manage.
For widespread hypertrophic scars, typically from severe burns, extensive trauma, infections, or large skin grafts, early and sustained application of silicone and compression therapy is crucial, initiated as soon as the wound is closed. Adjunctive therapies like blomycin, 5-fluorosyl, 5-FU, and verapamil have been explored. A pilot study even showed that an ointment containing losartan could significantly decrease the vascularity and pliability of scars and improve overall scar quality. Laser therapy, specifically pulsed dye laser and CO2 laser, is a very useful adjunct to reduce scar thickness, resurface texture, and treat redness or hyperpigmentation. Early surgery, using skin flaps for critical areas like the neck or armpit to release burn contractures, is indicated for functional impairment, and serial excision or tissue expansion can be used for very large, widespread scars to gradually remove them or prepare for grafts.
And for keloids, which are known to be far more resistant, what are the primary treatment approaches given their stubborn nature and tendency to recur?
For keloids, first-line treatments typically combine intralesional corticosteroid injections with silicone dressings and pressure therapy. Adjunctive agents like intralesional 5-FU, glomycin, and verapamil are used according to specific protocols. Studies have shown that 5-FU combined with corticosteroids is often more effective than either alone. For refractory keloids, defined as those still problematic after 12 months of consistent therapy, surgical excision combined with adjuvant therapy is often necessary. Excision alone has a high recurrence rate, from 50% to 100%, and carries the risk of the keloid actually growing larger.
So surgery alone is often not enough.
Rarely. Therefore, immediate post-operative adjuvant therapy, such as electron beam irradiation or brachytherapy, internal radiation, is used to significantly reduce recurrence rates, often by 50% to 95%. While there's a theoretical long-term association with radiation damage malignancy, current medical literature doesn't show a statistically significant link in the doses used. Studies comparing surgical excision with triamcinolone versus radiation therapy have shown similar recurrence rates, around 15%. Combined approaches, like surgical excision with intraoperative autologous platelet-rich plasma and postoperative radiation, have shown very low recurrence rates for keloids on the ear, for example. Other potential therapies with more limited evidence include cryotherapy, topical immune modulators, and various drugs that target growth factors or signaling pathways. The key is that there is no single best therapy for keloids, reflecting the incredibly complex and often genetically influenced nature of these scars. Future treatments may be more specifically tailored based on their distinct histological and molecular differences.
Now, moving beyond the scar itself, let's talk about chronic non-healing wounds. These are a major healthcare burden in many societies. What defines them, and why are they such a persistent problem? They seem to just linger.
By definition, chronic wounds are those that have failed to proceed through an orderly and timely reparative process to achieve anatomic and functional integrity over a period of 3 months. The prevalence is staggering. Millions of diabetic foot ulcers, chronic leg ulcers from venous insufficiency, and pressure ulcers occur annually in the U.S. alone. These numbers are unfortunately rising due to an aging population and increasing risk factors like diabetes and smoking. They represent an immense burden on healthcare systems and significantly reduce patients' quality of life, often leading to social isolation and significant pain.
What's happening at a biological level in these chronic wounds that makes them stall? Why can't the body just fix them like it normally does?
Their pathogenesis is a perfect storm resulting from systemic factors within the patient like malnutrition, aging, generalized tissue hypoxia, low oxygen, and conditions like diabetes, combining with localized factors at the wound site to create a profoundly hostile healing environment that overwhelms the body's normal processes. Chronic wounds show significant derangements in all healing stages, with abnormally elevated or depressed levels of cytokines, growth factors, and proteinases. For instance, fluid from chronic wounds has consistently higher levels of pro-inflammatory cytokines, like IL-1, IL-6, and TNF-alpha, compared to fluid from acute wounds. There's often an inverse relationship between these pro-inflammatory signals and essential growth factors like EGF and PDGF. Chronic wounds are also characterized by powerful, persistent pro-inflammatory stimuli, including bacterial colonization, necrotic dead tissue, foreign bodies, and localized tissue hypoxia. Tissue edema is also significant, increasing the distance for oxygen and nutrients to diffuse to cells. These wounds typically have high numbers of neutrophils expressing reactive oxygen species, ROS, those powerful free radicals, along with excessive proteases, creating a highly pro-oxidant damaging environment. This disturbed oxidant balance is a key factor amplifying and perpetuating inflammation. ROS can activate signaling pathways that control the expression of those pro-inflammatory chemokines and cytokines, as well as proteases. Bacterial components from colonization can also upregulate this chronic inflammatory response.
So an excessive inflammatory response that just won't quit, combined with this damaging chemical environment. And the extracellular matrix, the scaffold, is also out of balance.
Yes. Normal ECM is maintained by a dynamic balance of synthesis, deposition, and degradation. Chronic wounds exhibit a profound, unbalanced activity that overwhelms the tissue's protective mechanisms. While activated skin cells, fibroblasts, and endothelial cells can increase protease expression, the incoming neutrophils and macrophages are the primary source of many powerful proteases like cathepsin G and neutrophil elastase. Specific matrix metalloproteinases like gelatinases, MMP2, NACTR9, collagenases, MMP1, NACTR8, and stromolysins are all significantly upregulated in chronic venous ulcers, for instance. Pro-inflammatory cytokines induce MMP expression in chronic wounds and reduce the expression of their natural inhibitors, TIMPs, leading to a relative excess of MMP activity. This excess degrades or inactivates protective proteins like alpha-1 proteinase inhibitor, fibronectin, and others. Even crucial growth factors like PDGF and VEGF, vital for healing, are targeted and broken down by this excessive protease activity.
Beyond this protease imbalance, are there other proposed reasons for why a wound might become chronic? Other factors contributing to the stall?
Yes. Other factors include hyperproliferation of skin cells, keratinocytes, at the wound periphery, which can paradoxically inhibit fibroblast and keratinocyte migration and their natural programmed cell death. Fibroblasts in chronic wounds often show altered morphologies, slower proliferation rates, and reduced responsiveness to growth factors, meaning they're just not doing their job as effectively. The ratio of certain immune cells, like CD4-CD8 cells, is significantly lower in chronic wounds, which is particularly relevant in diabetic ulcers. And important growth factors like FGF, EGF, and TGF-beta are often reduced, likely due to that excessive protease degradation or being trapped by ECM molecules. Finally, there's increasing evidence that resident cells in chronic wounds undergo phenotypic changes that render them senescent, meaning they age prematurely, impairing their capacity for proliferation and movement.
And chronic wounds can lead to even more severe and dangerous complications, correct? It's not just about the wound itself.
They absolutely can. Chronically inflamed wounds are susceptible to neoplastic transformation, meaning they can become cancerous. Squamous cell carcinoma, for example, was initially described in chronic burn scars by Marjolin, hence the term Marjolin's ulcer. Chronic osteomyelitis, pressure sores, venous stasis ulcers, and hidradenitis can also develop neoplastic change. Any clinically atypical chronic wound, or one that looks suspicious, should be biopsied to rule out conditions like pseudoepitheliomatous hyperplasia, which is a premalignant condition, or outright squamous cell carcinoma. This is a serious concern that demands attention.
Let's quickly touch on some other specific systemic causes of abnormal wound healing that are vital for professionals to recognize. Hypoxia, for example, low oxygen levels. How big a deal is that?
Hypoxia, or insufficient molecular oxygen, is a fundamental inhibitor of healing. Molecular oxygen is absolutely essential for proper collagen formation. Ischemia, from conditions like heart failure or arterial disease, or even simply excessive wound tension, prevents adequate local tissue perfusion, meaning blood flow. While the body can try to initiate collagen synthesis under hypoxic conditions, molecular oxygen is critical for the post-translational hydroxylation of proline and lysine amino acids in collagen, which is necessary for the proper triple helix formation and cross-linking of collagen fibrils. Without enough oxygen, the collagen just isn't strong enough. Mild hypoxia can actually stimulate angiogenesis, which is beneficial, but collagen fibril assembly proceeds poorly when the partial pressure of oxygen, PO2, drops below 40 mmHg. While the wound periphery might have optimal PO2, the center often remains chronically hypoxic. Anemia, which is linked to overall reduced oxygen delivery, also results in altered wound histology, like decreased white blood cell infiltration, angiogenesis, and fibroblasts and collagen production, ultimately leading to delayed wound healing.
And what about tobacco smoking? How does that common habit impact wound healing? We know it's bad, but what are the specifics?
Tobacco smoking profoundly impairs healing in multiple ways. It causes peripheral vasoconstriction, narrowing blood vessels, and reducing wound blood flow by a significant 30–40%. Elevated carbon monoxide in smokers' blood inhibits enzyme systems essential for oxidative cellular metabolism, meaning cells can't produce energy efficiently. Nicotine also inhibits a chemical called platelet prostacyclin, which promotes platelet stickiness, leading to microscopic blood clots in tiny vessels and thus tissue ischemia. At a cellular level, tobacco use inhibits endothelial cell function, fibroblast function, nitric oxide synthase activity, which is vital for blood flow and healing, VEGF production, fibroblast proliferation, collagen synthesis, and even depletes vitamin C levels. It's a systemic assault on the healing process.
Wow, it hits almost everything.
It really does. Clinicians generally recommend complete smoking cessation for 4 to 6 weeks before elective surgical procedures to significantly optimize healing outcomes.
Diabetes mellitus is another huge global health challenge, affecting millions. How does it impair healing in such a complex way? It seems to affect multiple systems.
Diabetes mellitus impairs wound healing through multiple interconnected mechanisms. It causes both large vessel occlusion and microangiopathy damage to tiny blood vessels, leading to widespread tissue ischemia and increased susceptibility to infection. Diabetic sensory neuropathy, nerve damage that reduces sensation, predisposes patients to repeated trauma and unrelieved pressure on their feet, often leading to chronic ulcers. Tissue hypoxia is common, shown by reduced transcutaneous oxygen tension in diabetic limbs. The thickened capillary basement membrane further decreases microenvironmental perfusion, and increased capillary leak is often observed. A critical issue is VEGF dysregulation. The crucial angiogenic growth factor, VEGF, which is normally upregulated by hypoxia, is impaired in diabetic patients, meaning their cells don't respond to low oxygen by creating new blood vessels as they should.
So the normal signals aren't getting through, or they're not being generated properly, even when they're desperately needed for repair.
Correct. Diabetics are also uniquely susceptible to infection due to an attenuated inflammatory response, meaning their immune system is less effective. They have impaired chemotaxis. Immune cells don't move effectively to the infection, and inefficient bacterial killing. Infection further burdens their already tenuous blood supply, dramatically amplifying the risk of tissue necrosis and further damage. Lymphocyte and leukocyte functions are impaired. Collagen degradation is increased, while new collagen deposition is impaired, and existing collagen becomes brittle due to excessive glycosylation in the ECM sugar molecules inappropriately attaching to proteins. This glycation also diminishes the essential focal adhesion between fibroblasts and the matrix, decreasing fibroblast migration. Chronic hyperglycemia leads to increased advanced glycation end products, AGEs, which induce inflammatory molecules like TNF-alpha and IL-1 and actively interfere with collagen synthesis. High glucose levels also alter cell morphology, decrease cell proliferation, and cause abnormal skin cell differentiation. The early phase of healing is impaired by decreased immune cell function. Later, altered leukocyte infiltration and changes in wound fluid cytokines are observed. Important growth factors like FGF, EGF, and TGF-beta are often reduced, likely due to that excessive protease degradation or trapping by ECM molecules. Elevated MMPs and reduced TIMPs, similar to what we discussed in chronic wounds, are also prevalent. Finally, resident cells in chronic diabetic wounds often undergo cellular senescence, impairing their proliferation and movement. Recent literature even points to epigenetic alterations, like microRNA expression patterns which derail the inflammatory response in diabetic wounds.
And what about the devastating effects of ionizing radiation on wound healing? For patients undergoing cancer treatment, for instance.
Ionizing radiation primarily affects rapidly dividing cells, particularly during specific phases of the cell cycle. Injury to skin cells, keratinocytes, and fibroblasts directly impairs epithelialization and the formation of granulation tissue. Endothelial cell injury damage to blood vessel linings leads to inflammation of the vessel walls, atrophy, fibrosis, and ultimately delayed tissue repair. Repetitive radiation injury often causes repetitive inflammatory responses and ongoing dysfunctional cellular regeneration. Early side effects include redness, dry skin, hyperpigmentation, and hair loss. Late effects include skin thinning, dryness, tiny visible blood vessels, discoloration, fibrosis, and chronic ulceration. Both the inflammatory and proliferative phases of healing are profoundly disrupted. Cytokines like TGF-beta, VEGF, TNF-alpha, IF-10-gamma, IL-1, and IL-8 are often overexpressed after radiation injury, leading to uncontrolled matrix accumulation and fibrosis. Nitric oxide, which is important for collagen deposition and blood flow, is decreased in irradiated wounds, potentially explaining impaired wound strength. MMP1 is also decreased, contributing to inadequate soft tissue reconstruction. Skin cells in irradiated wounds show altered protein expression, and in non-healing ulcers, they display decreased expression of many crucial growth factors. Conversely, MMT2, negative-12, and Medicare-13 are elevated in irradiated skin cells and fibroblasts. Fibroblasts in irradiated tissue tend to generate disorganized collagen bundles due to dysregulation of MMPs and their inhibitors, TIMPs. TGF-beta, a key regulator of MMPs and TIMPs, is particularly relevant in radiogenic ulcers.
So radiation essentially throws the entire system off balance, creating a very difficult wound environment. How are these wounds typically treated? What approaches help?
Strategies for problematic radiogenic ulcers include meticulous standard wound care, advanced therapies like negative pressure wound therapy, aggressive nutritional optimization, and strategies to improve blood and oxygen delivery to the tissue. Hyperbaric oxygen therapy, or HBO, can significantly improve tissue oxygen partial pressure and promote the growth of new blood vessels, particularly in cases of bone death due to radiation. Clinical trials support HBO's efficacy as an adjunct to standard wound care for recalcitrant diabetic and radiation-induced wounds. Recent research also shows promise with adipose-derived stem cells injected into irradiated TU models in animals.
What about aging? How does simply getting older affect our body's amazing ability to heal? Does everything just slow down?
Older patients are indeed more prone to delayed healing and surgical wound breakdown. The aging epidermis has fewer of certain immune cells and pigment cells, and a flattened junction with the dermis. Keratinocyte proliferation is reduced, increasing the time it takes for skin cells to turn over by 50%. The dermis itself has fewer fibroblasts, macrophages, and mast cells, reduced vascularity, and less collagen and gags. There's a quantitative imbalance between collagen production and degradation, and a qualitative alteration of remaining collagen, which becomes more disorganized. Skin elasticity decreases due to altered elastin morphology. Furthermore, diminished light touch, pressure, and pain receptors, along with dermal atrophy, increase susceptibility to injury in older individuals. Immunosenescence, meaning reduced function of certain immune cells and fibroblast activity, impairs healing and increases the likelihood of chronic wounds. Microvascular disturbances predispose to ischemic ulcers, and there's reduced oil secretion and vitamin D3 production in the skin. ECM dysregulation, including altered MMPs, decreased reepithelialization, depressed collagen synthesis, impaired angiogenesis, and decreased pro-angiogenic growth factors, are all consistently observed in elderly animal studies. Impaired macrophage activity with reduced phagocytosis and delayed infiltration, as well as impaired B lymphocyte activity, have also been demonstrated. Promisingly, recent animal studies suggest that anti-aging agents like metformin and resveratrol, acting through specific cellular energy pathways, may offer potential benefits for wound healing in the elderly. This is a fascinating area of research for future treatments.
And malnutrition seems like an obvious factor, but what are the specifics of how it impacts healing? Which nutrients are most critical?
Protein catabolism, meaning the breakdown of protein, significantly delays wound healing and promotes wound breakdown, particularly when serum albumin levels, a key indicator of protein status drop below 2.0 grams per deciliter. This is fortunately reversible with protein supplements. Vitamin deficiencies also critically impact healing, often by acting as essential cofactors for enzymes. Vitamin C deficiency, for example, can delay healing due to its indispensable role in proline hydroxylation, which is essential for collagen stability. Vigorous supplementation can reverse this. Vitamin A deficiency impedes the activation of monocytes and the deposition of fibronectin, affecting cellular adhesion, and impairs TGF-beta receptors. It can also counteract the negative effects of glucocorticoids. Vitamin K deficiency limits the synthesis of crucial blood clotting factors, and this can be exacerbated by certain antibiotics, so checking clotting parameters is important. Zinc is a necessary cofactor for enzymes involved in RNA and DNA synthesis, and its deficiency impairs early wound healing, though it's rare except in severe cases like large burns or liver disease. Iron deficiency anemia is a debatable cause of delayed healing, but ferrous ion is needed for proline to hydroxyproline conversion. The general recommendation for optimal healing is always a well-rounded diet with adequate protein, sufficient caloric value, and appropriate vitamin and mineral supplementation.
Finally, certain commonly administered drugs can also impede healing. What are the main culprits a professional needs to be aware of that might be slowing things down?
Exogenously administered drugs can indeed directly impair wound healing. Chemotherapeutic agents such as doxorubicin, nitrogen mustard, cyclophosphamide, and methotrexate are potent inhibitors. They interfere with uncomplicated wound healing by reducing the proliferation of key mesenchymal cells, reducing platelet and inflammatory cell counts, and decreasing the availability of crucial growth factors, especially if given before a surgical procedure. Tamoxifen, an anti-estrogen drug, decreases cellular proliferation and causes a dose-dependent decrease in wound-breaking strength, possibly due to decreased TGF-beta production. Glucocorticosteroids like prednisone significantly impair fibroblast proliferation and collagen synthesis, leading to decreased granulation tissue formation. They also stabilize lysosomal membranes. More recently, studies have shown that steroids inhibit keratinocyte migration and proper wound healing by activating specific cellular signaling pathways. Vitamin A, notably, can partially reverse some of these steroid effects. The diminished wound-breaking strength caused by exogenous steroids is both time and dose-related. High bases of nonsteroidal anti-inflammatory drugs, or NSAIDs, have been reported to delay healing, but doses in the therapeutic range are generally unlikely to have a significant impact on uncomplicated wound healing.
So we've identified all these intricate mechanisms of healing and what can go wrong systemically or locally. Now, let's pivot to the treatment of chronic wounds. What are the main challenges here for medical professionals, and why have traditional approaches sometimes fallen short? These wounds seem particularly stubborn.
The management of chronic wounds is incredibly challenging. It's slow, labor-intensive, and extremely expensive, with no guarantee of healing unless all local and systemic factors are comprehensively addressed. Early wound healing research identified key structural proteins and signaling molecules, leading to the logical step of applying topical growth factors directly to chronic wounds. This was very promising in theory, as it aimed to replace missing factors. However, these topical growth factors almost universally failed to produce clinically significant improvements in patients.
Why did these seemingly promising topical growth factors fail? What was the missing piece? It seems like a logical approach.
The presumed reason for their widespread failure was their rapid degradation by the excessive proteases present in the hostile fluid environment of chronic wounds. This was a critical lesson and highlighted the complex nature of wound healing. Simply replacing one missing element isn't enough when the entire wound microenvironment is dysfunctional and actively breaking down beneficial molecules. It underscored that we need more comprehensive approaches that address the overall balance.
That brings us to skin substitutes, which have become a major advancement in addressing these complex wounds. How do they work, and what advantages do they offer over just, say, a traditional graft?
Skin substitutes are ingeniously designed to provide multiple factors that collectively alter the wound microenvironment in favor of healing. They address the limitations of traditional autologous skin grafts, where skin is harvested from the patient themselves. While effective, autografts have drawbacks like donor site pain, scarring, and limited availability, especially for large wounds. Biologic skin substitutes like cadaveric allografts or porcine bovine xenografts have been used for years but are temporary. They don't integrate permanently and are eventually rejected by the host immune system.
So what's their primary mechanism of action beyond just simple wound coverage? How do they help the healing?
They provide crucial wound coverage, which immediately creates a more favorable healing environment by excluding pathogenic bacteria and limiting dangerous fluid loss. But more profoundly, they provide essential components like extracellular matrix proteins, living cells such as keratinocytes and fibroblasts, and a range of bioactive molecules like cytokines, chemokines, and growth factors. This facilitates wound repair through a process called dynamic reciprocity, where the substitute actively interacts with and educates the wound bed. These products also have significant economic impact, potentially saving millions annually by reducing dressing costs and treating wound-induced complications, especially for chronic wounds like venous, diabetic, and pressure ulcers, which account for a vast majority of all chronic wounds. Their design varies greatly depending on the skin layer they're designed to replace, epidermal, dermal, or bilayer. Epidermal replacements are made from cultured patient keratinocytes and are often fragile. Dermal substitutes are structural 3D matrix materials that mimic the ECM, often incorporating cells or bioactive molecules to rebalance the wound microenvironment. Bilayer materials combine features of both.
Can you give us a few specific examples of these different types of skin substitutes? What's out there?
Certainly. One of the first major breakthroughs was Integra, a bilayer substitute with a silicone outer layer and a deeper layer made of bovine cartilage and jays. The silicone layer is removed after a few weeks, and a thin, split-thickness skin graft is then applied over the integrated dermal component. It's used for burn wounds and can be applied directly over exposed tendon and bone. A more recent classification system, proposed in 2018 by Davison-Kotler and colleagues, categorizes them by their cellularity, layering, the skin region they replace, the material they're made from, and their permanence. For example, alloderm is a cellular, single-layer, temporary dermal substitute derived from cadaveric human skin used extensively in breast reconstruction. BioBrain is a cellular bilayer material composed of silicone, nylon mesh, and porcine collagen, used for full-thickness wounds and removed once the wound heals. Epicel is an epidermal-only cellular product derived from the patient's own keratinocytes. A major drawback of many cultured autografts generally is that they are extremely thin, highly susceptible to shearing forces, and prone to significant contraction after application.
And there are exciting new advances in burn treatment, combining these substitutes with stem cells. That sounds very promising.
Yes, this is a truly promising area. Researchers have found that skin debrided from burn patients can yield living cells, including those with mesenchymal stem cell characteristics, which can then be cultured and expanded. These cells can then be embedded in a specialized skin scaffold and used to treat wounds in animal models, promoting angiogenesis and reepithelialization without negative side effects. Combining stem cell therapy with skin substitutes holds immense promise for more individually tailored wound care in the future, offering a highly personalized approach to healing.
Speaking of wound care, dressings have also evolved significantly beyond just simple gauze. What makes an ideal dressing today, and what are the key concepts guiding their selection? There are so many options now.
They certainly have. Wound dressings have been around since antiquity, but saw little evolution until Joseph Lister introduced antiseptic dressings in 1867. The ideal modern dressing should create a moist, clean, and warm environment, effectively remove excess fluid, prevent the wound from drying out, allow for proper gaseous exchange, be impermeable to harmful microorganisms, provide thermal insulation, prevent particulate contamination, be non-toxic, offer mechanical protection, be non-traumatic upon removal, be easy to use, and importantly, be cost-effective.
So the choice of dressing depends heavily on the wound itself and its needs, how much fluid it's producing, for instance. What are the key concepts that guide this selection process?
The two critical concepts are occlusion and absorption. Studies show that epithelialization, the resurfacing of the skin under a moist occlusive dressing, is twice as fast as an uncovered wound allowed to dry out and scab. An occlusive dressing creates a mildly acidic pH and low oxygen tension in the wound, which is conducive for fibroblast proliferation and granulation tissue formation. However, a crucial caution for you. Without specific antimicrobial properties, a purely occlusive dressing can paradoxically worsen infection by promoting bacterial overgrowth.
Okay, so moisture is good, but it can trap bacteria if you're not careful.
Exactly. For wounds with significant fluid discharge or exudate like many chronic leg ulcers that can produce a lot of fluid, an absorptive dressing is needed to prevent maceration, softening and breakdown of the surrounding skin, and to help reduce bacterial load.
How are modern dressings categorized for professionals today? What are the main types?
They're broadly categorized into 4 classes: non-adherent fabrics, occlusive dressings, absorptive dressings, and creams, ointments, and solutions. Non-adherent fabrics are fine mesh gauzes, often impregnated with substances to improve their properties, like scarlet red for skin graft sites, or Xeroform, which contains bismuth tribromophenate for mild antimicrobial action.
And occlusive dressings are key for maintaining that moist healing environment you mentioned earlier.
Yes. They provide moisture retention, mechanical protection, and a bacterial barrier. This can be non-biologic, such as clear polyurethane films like Tegaderm, which are waterproof and allow visualization but are non-absorptive and can leak, or hydrocolloids like Duoderm, which absorb exudate to form a moist gel. Alginates, derived from seaweed, convert into a hydrophilic gel on contact with exudate, providing excellent absorption. Hydrogels, mostly water, are primarily rehydrating for dry wounds, offering little absorption. Biologic occlusive dressings include cadaveric human skin allografts, animal-derived xenografts like pig skin, amnion from human placentas, and various engineered skin substitutes we just discussed. Allografts and xenografts are temporary and will be rejected if left on too long.
What about absorptive dressings for heavily exuding wounds, when you need to soak up a lot of fluid?
For wounds with high exudate, wide mesh gauze is the oldest, but it loses effectiveness quickly when saturated and can be painful to remove as it pulls away new tissue. Newer foam dressings, like Lyofoam, are hydrophobic polyurethane sheets that are highly absorbent and non-adherent, comfortable and easy to remove. However, it's important to note, healing under some absorptive dressings may be slower than occlusive ones, possibly due to accidentally wicking away beneficial cytokines from the wound bed or decreasing keratinocyte migration.
And finally, the wide category of creams, ointments, and solutions. This covers a lot.
It does. This is a broad category, from simple zinc oxide pastes to various powerful antibacterial preparations. Antibacterial options include acetic acid for specific bacteria, Dakin solution, iodine-containing solutions, silver nitrate, and topical antibiotics like mafenade and silver sulfadiazine, which are common for burns. These are generally used when there are clear clinical signs of infection or if quantitative cultures show a high bacterial count. Antibacterial ointments like bacitracin, neomycin, and polymyxin B-sulfate, often combined in products like polysporin or neosporin, are soothing, lubricate the wound, are reclusive, and offer about 12 hours of activity. Enzymatic debridement agents like sutilins, collagenase, and papain specifically remove necrotic tissue, helping to prepare the wound bed. Normal saline wet-to-dry gauze is an older method that removes necrotic and sometimes newly formed tissue and can be quite painful. The specific dressing option for non-infected clean wounds ultimately depends on the wound type. Incisional wounds may use simple 3-layer dressings or occlusives. Partial thickness wounds like abrasions or donor sites may use no dressing, impregnated gauze, creams, or occlusives. And complex full-thickness wounds like pressure sores might use specialized alginates, hydrogels, or advanced systems like negative pressure wound therapy.
Beyond dressings and skin substitutes, there are other cutting-edge advanced therapies for complex wounds that truly highlight how far the field has come. Let's start with hyperbaric oxygen therapy, or HBO. This involves breathing pure oxygen under pressure, right?
That's right. HBO therapy is essentially using oxygen as a powerful drug, with a hyperbaric chamber as the delivery system. The goal is to significantly increase the partial pressure of oxygen at the target tissue area. You see, wound ischemia, meaning insufficient blood flow and oxygen, is the single most common cause of wound healing failure. HBO therapy is used for a variety of conditions, including certain bacterial infections, decompression sickness, to improve skin graft and flap survival, treat acute burns, and, importantly for our discussion, chronic and hypoxic wounds, as well as complex issues like osteoradic and radiation injuries. There's increasingly strong evidence for its specific use in chronic diabetic ulcers and radiation-induced wounds.
So it's literally about flooding the tissue with oxygen, even where blood flow is poor, to overcome that deficit.
Yes. Ischemia, or tissue hypoxia, where the oxygen levels in the tissue drop below a critical threshold of about 30 mmHg, significantly impairs normal cellular metabolic activity, fibroblast proliferation, collagen synthesis, and epithelialization. HBO therapy involves inhaling 100% oxygen at elevated pressures, typically 1.9 to 2.5 times atmospheric pressure for specific periods. This can increase tissue oxygen levels tenfold, providing sufficient oxygen for metabolic requirements even when blood flow is compromised. This elevated oxygen level can last for 2 to 4 hours post-treatment. HBO also directly induces endothelial cell nitric oxide synthesis, which promotes blood vessel dilation and angiogenesis, the growth of new blood vessels. Oxygen itself stimulates angiogenesis, enhances fibroblast and leukocyte function, and helps normalize blood flow regulation in the skin. Recent animal studies even show that HBO increases stem cell proliferation, upregulates angiogenesis, and improves wound healing capacity in diabetic mice, and it has a synergistic effect when combined with stem cell therapy.
How do you know if a patient will truly benefit? And what's a typical treatment protocol like for chronic wounds? Sounds quite intensive.
Evaluating the underlying vascular supply is essential. If there are major arterial issues, revascularization restoring blood flow should ideally precede HBO. A key diagnostic tool is measuring transcutaneous oxygen pressure, or TCPO2, which assesses wound perfusion and oxygenation. A wound TCPO2 less than 35 mmol Hg in room air indicates a hypoxic wound, and importantly, an in-chamber TCPO2 of 200 mmol Hg or more during a test treatment suggests potential benefit. Treatments are typically delivered at 1.9 to 2.5 atmospheres for 90 to 120 minutes per session, with the patient breathing 100% oxygen, usually daily, 5 to 6 times per week. It is always an adjunct to surgical or medical therapies, not a standalone treatment. Clinical improvement is typically noted after 15 to 20 treatments.
What are the risks and potential complications of such high oxygen levels and pressures? Are there downsides?
Complications can arise from both the pressure changes and the elevated oxygen levels. Middle ear barotrauma, ranging from simple redness of the eardrum to perforation, is the most common pressure-related complication. Pneumothorax, a collapsed lung, especially tension pneumothorax, is far less common but life-threatening. From elevated oxygen levels, we can potentially see brain oxygen toxicity, manifesting as seizures, oxygen lung toxicity, ranging from tracheobronchitis to severe acute respiratory distress syndrome due to free radicals, and temporary vision changes like myopia. Absolute contraindications include uncontrolled pneumothorax and current or recent treatment with certain chemotherapy drugs like bleomycin or doxorubicin due to increased risks of cardiac and pulmonary toxicity.
What does the clinical evidence say about the effectiveness of HBO for chronic wounds? Is it well supported?
It's mixed but growing. For diabetic ischemic foot ulcers, older randomized controlled trials showed HBO could reduce amputation rates, although the more recent DAMA2CL study showed improved limb salvage and wound healing rates that were not statistically significant in their primary outcome. For chronic venous ulcers, a double-blind study showed HBO can improve refractory healing, but patient selection is key, focusing on those with hypoxic peri-wound margins after 4 weeks of high-quality standard wound care. A 2016 Cochrane review on chronic irradiated wounds concluded HBO improves outcomes in radiation injuries of the head, neck, anus, and rectum, and reduces osteoradionecrosis after tooth extractions, although these recommendations were based on smaller studies. A 2016 European Consensus Conference on Hyperbaric Medicine concluded HBO is indicated for open fractures with crush injury, specific types of osteoradionecrosis, soft tissue radionecrosis, diabetic foot ulcers, and femoral head necrosis, with weaker evidence for compromised skin grafts, flaps, ischemic ulcers, and refractory osteomyelitis. Despite potential benefits, HBO is costly, typically ranging from $15,000 to $40,000 for a course of treatment. It is generally reserved for carefully selected complicated cases rather than uncomplicated ulcers due to the cost and logistics.
Another significant advancement that has truly revolutionized wound management is negative pressure-assisted wound therapy, often known by brand names like VAC. This really changed the game, didn't it?
It truly did. NPWT was initially described by Argenta and Associates in 1997. It involves applying controlled subatmospheric pressure, essentially a vacuum to wounds. Clinicians observed it removed chronic edema, increased local blood flow, and powerfully stimulated the formation of granulation tissue. It's now widely used on acute, subacute, and chronic wounds, and has consistently shown significant improvement in wound depth, faster healing times, and fewer complications compared to traditional saline dressings.
The mechanism seems almost too simple for such profound results. Is it fully understood how a vacuum can do all that, just sucking fluid out?
The exact mechanism is still being actively determined, but we have many insights. Initially, it was hypothesized to be simply about removing excess fluid while keeping the wound moist. But studies have shown a remarkable 5-fold increase in blood flow to cutaneous tissues under NPWT. There's also an increase in capillary size and stimulated endothelial proliferation and angiogenesis. And here's a fascinating contradiction for you. Despite the improved healing, NPWT has sometimes shown increased bacterial counts, which suggests its primary mechanism isn't solely about direct infection control. This is a point that continues to intrigue researchers.
That is counterintuitive. Better healing, but more bacteria.
Yeah. Other research indicates NPWT produces three-dimensional mechanical stress within cells, called microstrain, and across the wound, called macrostrain, leading to increased cellular proliferation and a higher density of tiny blood vessels. Evidence also strongly suggests NPWT alters wound fluid composition by removing potentially deleterious proteinases and inflammatory cytokines like MMP1, MMP2, MMP9, and TNF-alpha, essentially cleaning up the bad actors in the wound microenvironment.
So it's cleaning up the bad stuff, keeping it moist, and stimulating the good stuff through mechanical stress and blood flow. What are the key clinical benefits we've seen from randomized controlled trials?
Randomized controlled trials have demonstrated numerous significant benefits. A decrease in wound volume and size, accelerated wound bed preparation and granulation tissue formation, overall accelerated healing, improved rates of skin graft take, decreased drainage time for acute wounds, reduced complications, enhanced response to first-line treatments, and even decreased cost. More recent data shows improved outcomes across a wide range of complex wounds, including diabetic wounds, ischemic ulcers, and complex surgical wounds in various specialties. Molecular analysis from a study by Karim and colleagues on diabetic non-healing ulcers showed that just 10 days of NPWT significantly downregulated pro-inflammatory cytokines and specific MMPs, while significantly upregulating crucial growth factors like VEGF, TGF1, and their inhibitors. This suggests NPWT actively rebalances the wound microenvironment, increasing growth factors, decreasing inflammatory cytokines, and normalizing MMP activity, thereby enhancing healing.
It's cost-effective too, which is a major bonus in healthcare. That's always important.
Yes, NPWT represents a significant improvement in cost effectiveness. Reports have shown up to a 78% decrease in hospital stay and a 76% decrease in cost for acute and chronic wounds, translating into substantial savings for healthcare systems. This has even facilitated earlier discharge and transition to home healthcare for many patients. A recent development is NPWT with instillation, which uses an open cell foam sponge to periodically instill sterile fluid to help remove thick exudate or infectious products. While some solutions include dilute antiseptics, studies have shown no significant difference in effectiveness between simple normal saline and antiseptic solutions. And portable, single-use vacuum-assisted closure devices are becoming increasingly popular for outpatient use and for more superficial or surgical incisional wounds, making this therapy more accessible.
This brings us to a truly unique and fascinating area that has profound implications for all of us. Fetal wound healing, often referred to as the scarless ideal. What makes it so fundamentally different from adult healing? It sounds almost miraculous.
Fetal skin wounds heal rapidly, completely without the scarring and inflammation that characterize adult skin wounds. In adult cutaneous healing, dermal appendages like hair follicles, sweat glands, and sebaceous glands fail to regenerate. They are simply replaced by scar tissue. But in fetal healing, these structures do regenerate, restoring true tissue architecture. Healed adult wounds have densely packed collagen bundles oriented perpendicularly to the wound surface, creating a noticeable scar, while collagen in uninjured and fetal skin retains a natural, more relaxed reticular pattern. Fetal wounds also reepithelialize faster, with less new blood vessel formation and a faster increase in wound strength. The profound goal of studying fetal wound healing is to understand this ideal tissue repair to provide us with crucial insights and tools to regulate and control adult wound healing processes, hopefully minimizing scarring.
So it's not just about being a fetus. Gestational age and wean size play a role in whether it truly heals scarless. It's not guaranteed.
Absolutely, it's not a universal guarantee throughout fetal development. Scarless repair is highly dependent on both the gestational age and the specific wound size. There's a wound size threshold, typically 6-10 millimeter diameter for earlier gestation, around 60-70 days, and 4-6 millimeter for later gestation, 80-90 days, above which scars can begin to form. Larger wounds tend to extend the healing time and expose the tissue to a different extracellular matrix and growth factor profile, which may actually stimulate the formation of myofibroblasts and lead to scarring. The transition from scarless to scarring repair in human skin typically occurs at the beginning of the third trimester. And overall, fetal wounds heal faster than neonatal wounds, which in turn heal faster than adult wounds, demonstrating a clear developmental progression in healing capacity.
And the regeneration of skin appendages, hair follicles, and glands, that's a huge difference from adult healing. Why is that significant? What does it tell us?
It is indeed. Dermal fibroblasts actually play a crucial role in inducing the overlying epithelium to form these complex structures like hair follicles or glands. Wounds created early in gestation heal without scarring and with the regeneration of dermal appendages, truly suggesting complete tissue regeneration, not just repair. In contrast, late gestation wounds heal with scarring and without dermal appendages. This transition strongly suggests that fetal fibroblasts gradually lose their ability to induce appendage formation as gestational age advances, moving towards an adult-like healing response.
Is it something inherent to the fetal environment, like the amniotic fluid, or something intrinsic to the fetal cells themselves that makes this ideal healing possible? Or both?
It's likely a combination of both. There are extrinsic factors, like the unique amniotic fluid environment, and equally important intrinsic factors, like differences in cellular receptors and growth factor expression within the fetal tissues. The fetal environment is characterized by sterile, hyaluronic, acid-rich amniotic fluid, which actively contributes to a decreased inflammatory response. The increased number of hyaluronic acid receptors on fetal cells and the sheer abundance of hyaluronic acid itself may create a permissive environment that facilitates rapid fibroblast movement, leading to the increased rate and efficiency of fetal healing.
But the intrinsic factors, the properties of the fetal cells themselves, seem to be the focus of more recent and exciting research. What's special about fetal fibroblasts compared to adult ones?
Much research focuses on fetal fibroblasts because they possess characteristics quite different from adult fibroblasts. For instance, proline hydroxylation, a crucial rate-limiting step in collagen synthesis, is increased in early gestation fetal human fibroblasts. This activity gradually decreases to adult levels after 20 weeks of gestation. Fetal wounds also show an earlier appearance of various collagen types and a greater ratio of type 3 to type I collagen, which is consistent with the higher type 3 collagen found in normal fetal tissue, contributing to its unique properties. Fetal fibroblasts show significantly higher collagen production in vitro compared to adult counterparts, possibly due to a unique regulatory mechanism for prolyl hydroxylase, explaining their higher activity in fetuses under 20 weeks. Collagen synthesis naturally decreases to adult levels after 20 weeks, and interestingly, collagen degradation increases with gestational age. The increased gene expression of certain MMPs, like MMP1 known of 3 and NR9, also correlates with the onset of scar formation in non-wounded fetal skin, suggesting that later gestation fetal skin undergoes a more adult-type tissue remodeling process.
So the extracellular matrix composition itself is different in fetal wounds. Beyond just the hyaluronic acid, the whole environment is different.
Yes, hyaluronic acid consistently remains elevated in fetal wounds, and its decreasing levels during gestation correlate directly with an increasing scarring potential. This unique ECM composition, particularly the abundance of hyaluronic acid, may fundamentally influence how collagen fibrils are deposited, leading to the characteristic loose collagen pattern seen in healed fetal wounds, as opposed to the dense, disorganized collagenous pattern in adult scars. However, it's worth noting that few studies have successfully modified ECM components in adult wounds to truly replicate this fetal pattern.
And the inflammation is dramatically reduced in the fetus. Is that a major reason for scarless healing? The lack of that initial fierce reaction?
Indeed, the fetus exhibits a dramatically reduced inflammatory response. You see a striking lack of neutrophil infiltration and decreased endogenous immunoglobulins, key components of the adult immune response. The relative scarcity of macrophages and profound differences in their temporal appearance in fetal wounds are thought to explain the distinct growth factor profiles and the dramatically reduced inflammation. Studies have shown a direct correlation between increased macrophage recruitment in older fetuses and the development of increased scarring, suggesting that inflammatory cells play a direct role in scar formation.
And the growth factors, those crucial signaling molecules, are also very different in the fetus. The signals themselves are dialed down.
Yes, the growth factor profile is markedly distinct. Fetal wounds have notably decreased levels of TGF-beta and FGF2. TGF-beta is the most studied growth factor in fetal wound repair because its production may be blunted in hypoxemic conditions. This has led to the theory that the decreased oxygen tension in the fetal environment might directly inhibit TGF-beta production, thus leading to decreased scar formation. The differential expression of TGF-beta isoforms is also important, with some research suggesting that TGF-beta may actually limit scarring, unlike its counterparts. PDGF also disappears more rapidly in fetal wounds. This overall paucity of growth factors in fetal wounds is often attributed to that decreased inflammatory cell recruitment. It's a fascinating evolutionary trade-off. It's hypothesized that the normal adult-type inflammatory response evolved to quickly reduce the risk of infection after injury, but it comes at the expense of healing quality, leading to scarring. Unfortunately, attempts to manipulate growth factors to make adult wounds fetal-like have generally failed to achieve completely scarless healing or dermal appendage regeneration, highlighting the complexity. Other factors like IGF, EGF, and various interleukins have also been proposed to play a role, but the balance is critical.
What about the role of Wnt signaling and those specialized myofibroblasts in fetal healing? Are they absent or different?
Wnt signaling plays a profound role in embryogenesis and is involved in multiple phases of wound healing in adults. This pathway is deeply involved in tissue remodeling and repair, and when dysfunctional, it contributes to scarring. Studies have identified a crucial link between Wnt signaling and TGF-beta expression and function, suggesting that insights here could lead to novel therapeutic interventions for reduced scarring in adults. The presence of myofibroblasts and concurrent scar formation in adult healing also strongly suggests that the transition of fibroblasts into this contractile phenotype significantly contributes to scarring. In early gestation fetal lambs, excisional wounds showed a remarkable absence of scar formation and, critically, an absence of alpha-smooth muscle actin expression, the defining marker of myofibroblasts. This only appeared after 100 days gestation, along with the onset of scar formation, further supporting the idea that the absence of myofibroblasts is a key factor in scarless healing.
And finally, stem cells must play a role, even in fetal healing, given their regenerative potential. Are they more active or different in the fetus?
Absolutely. Recent findings highlight the crucial role of multiple pluripotent stem cells, including epithelial stem cells, mesenchymal stem cells, MSCs, and even rare circulating small dot cells. Epithelial stem cells interspersed in the basal layers of the skin can migrate to the epidermis after injury and differentiate into new skin cells. Mesenchymal stem cells, or MSCs, sourced from places like placenta, bone marrow, and fat, play diverse roles in regenerative healing, immunomodulation, calming the immune system, antifibrosis, anti-apoptosis, promoting angiogenesis and preventing excessive inflammation, often by secreting anti-inflammatory signals like IL-10. Those intriguing small dot cells are 20 times more abundant in fetal blood, and when transplanted into postnatal mouse wounds, they migrated to the wound bed and significantly decreased scarring. Further investigation of these stem cell populations is absolutely crucial. Finally, cellular senescence, where cells lose their ability to replicate after stress but remain metabolically active, is now believed to significantly influence wound healing and tissue remodeling, and may help explain the differences between fetal and adult wound healing. Further investigations into all these complex mechanisms are vital to translate this profound understanding into clinical applications for improved, less scarring healing adults.
Building on that incredible deep understanding of the body's natural processes, tissue engineering represents our attempt to create biological substitutes to help wounds heal better, and perhaps even achieve a closer approximation to that scarless ideal we just discussed. How is tissue engineering defined in this field?
The National Science Foundation in 1987 famously defined tissue engineering as the application of the principles and methods of engineering and the life sciences toward the development of biologic substitutes to restore, maintain, or improve function. In wound healing, this primarily translates to the creation of advanced skin products, which can be made of cells, extracellular matrix components, or powerful combinations of both. This field has progressed rapidly, mainly driven by the inherent limitations associated with traditional autografts, like the pain and scarring at the donor site from where the skin is taken. These new engineered skin substitutes aim to carefully mimic native tissues to promote sustained healing without rejection. The use of biological dressings, scaffolds, and even integrating stem cell therapy and gene therapy all fall under the sophisticated umbrella.
So it's about making a better, often temporary, or even permanent skin patch that's biologically active, not just a simple cover.
Precisely. Bioengineered skin substitutes have the potential to save millions of dollars annually in healthcare by significantly reducing spending on dressings and treating wound-induced complications, particularly for chronic wounds like venous, diabetic, and pressure ulcers, which count for a vast majority of all chronic wounds. They serve a crucial dual function, acting as protective dressings by limiting bacterial colonization and fluid loss, while also actively stimulating healing through their interaction with the wound bed. Their design varies greatly depending on which layer of skin they're designed to replace. Epidermal, dermal, or a bilayer combining both. Epidermal replacements are typically cultured patient keratinocytes attached to a carrier, but they can be quite fragile. Dermal substitutes are structural 3D matrix materials that truly mimic the extracellular matrix, often incorporating cells or bioactive molecules to rebalance the wound microenvironment. Bilayer materials combine features of both, aiming for a more complete restoration.
What are some examples? And are there any downsides or limitations to these advanced substitutes? They sound amazing, but what are the catches?
They're used for both acute and chronic wounds, but there's still no single ideal substitute, and cost can be a significant factor. One of the early major breakthroughs was Integro, a bilayer substitute with a silicone outer layer and a deeper layer made of bovine cartilage and jays. The silicone layer is removed after a few weeks, and a thin, split-thickness skin graft is then applied over the integrated dermal component. It's used for severe burn wounds and can be applied directly over exposed tendon and bone. A more recent classification system from Davison Kotler and colleagues in 2018 categorizes them by their cellularity, layering, the specific skin region they replace, the material they're made from, and their permanence. For example, alloderm is an acellular, single-layer, temporary dermal substitute derived from cadaveric human skin, used extensively in breast reconstruction. BioBrain is an acellular bilayer material composed of silicone, nylon mesh, and porcine collagen, used for full-thickness wounds and removed once the wound heals. Epicel is an epidermal-only cellular product from the patient's own keratinocytes. A major drawback of many cultured autografts generally is that they are extremely thin, highly susceptible to shearing forces, and prone to significant contraction after application, making them challenging in certain areas.
And building on that, what about directly manipulating genes and cells for healing? Gene and stem cell therapy sounds like the ultimate in precision healing, maybe even getting closer to regeneration.
It's truly the cutting edge. In gene therapy, powerful tools like the CRISPR system, originally a bacterial defense mechanism, have the potential to precisely target human genes that encode crucial growth factors and cytokines involved in wound healing. The exciting idea is to specifically upregulate these beneficial genes in an inducible manner, meaning we can turn them on when needed, delivering precisely what the wound requires at the right time. This offers an unprecedented level of control.
And then there are mesenchymal stem cells, or MSCs, which we heard about in fetal healing. What's their broader role and potential as a therapeutic agent? They seem incredibly versatile.
MSCs are remarkable. They are multipotent, undifferentiated cells, meaning they can develop into various cell types, and they are found in diverse tissues like the placenta, bone marrow, and fat. They play incredibly major, multifaceted roles across all phases of wound healing. In the early inflammatory phase, days 1 through 3, they actively regulate inflammation, suppressing pro-inflammatory signals like TNF-alpha, producing anti-inflammatory molecules like IL-10 and IL-4, and even blocking excessive T-cell proliferation. In the proliferative phase, around 2 weeks, they produce crucial growth factors like VDGF, HGF, and PDGF, and actively recruit essential cells like keratinocytes, dermal fibroblasts, and even the body's own resident stem cells to the wound site. In the long remodeling phase, up to 2 years, they continue to play a role, producing factors like TGF-3-3F and regulating the balance of MMPs and TIMPs, thereby precisely influencing collagen deposition and ultimately improving scarring. Overall, MSCs coordinate inflammatory cell activity, inhibit harmful pro-inflammatory cytokines, stimulate the crucial process of engulfing debris, phagocytosis, and promote organized extracellular matrix deposition. Skin substitutes containing MSCs have been invaluable in characterizing the precise array of cytokines, growth factors, and chemokines needed for normal healing, which is helping us understand the whole symphony of signals.
So their therapeutic potential is enormous because they can influence so many different aspects of healing, from calming inflammation to promoting new tissue growth and even improving scar quality. They're like tiny doctors in the wound.
Exactly. MSCs can interact profoundly with surrounding cells, expressing the trophic factors, those supportive and nourishing molecules required for dermal healing in a highly regulated manner. This has the potential to significantly reduce the fibrotic burden of the wound and improve scarring. They can modulate T-cell biology, neutralize damaging reactive oxygen and nitrogen species, deliver powerful anti-fibrosis factors like HGF, inhibit excessive wound healing fibroblasts, promote robust angiogenesis, and even direct the differentiation of other cells within the wound. This makes them incredibly versatile for achieving truly precision healing, moving us closer to the regenerative ideal.
And beyond these well-established categories of advanced therapies, there are truly exciting new horizons in wound care, focusing on innovative treatments that directly stimulate cells and chemical mediators in ways we're only just beginning to fully understand, and things that sound almost sci-fi.
Yes, this includes therapies like electrical stimulation, therapeutic ultrasound, and vibration therapy. Electrical stimulation, for instance, capitalizes on the fascinating fact that wounds naturally carry a direct current electrical gradient. Applying external electrical currents can alter gene expression within cells, affecting the production of essential chemokines, cytokines, and collagen, thereby promoting a more favorable healing environment. It appears to activate specific intracellular pathways within cells. Experimental studies on human dermal fibroblasts have shown that electrical current stimulation can increase collagen and MMP1 levels, both important for remodeling. Clinically, specific types of pulsed currents have shown efficacy in accelerating healing. A pilot study in older adults with pressure wounds found significant improvements in pain and wound size when transcutaneous electrical nerve stimulation, TENS, was combined with standard care. It's believed that electrical stimulation subtly affects gene expression, with studies showing altered expression of over 100 genes in healthy volunteers exposed to it.
What about therapeutic ultrasound? How does that work its magic on wounds? Using sound waves for healing.
Kilohertz ultrasound uses sound waves to cause phenomena like microstreaming, the movement of fluids around vibrating cells and cavitation, which produces tiny bubbles that generate mechanical energy. This energy can alter the cell membrane and activate internal signaling pathways. It's believed to primarily affect the proliferative phase of wound healing, for instance, by increasing macrophage activity and collagen production. Low-frequency ultrasound has been shown to reduce the size of chronic venous leg ulcers. Non-contact low-frequency ultrasound used for venous, diabetic, and pressure wounds has also been shown to reduce bacterial burden and decrease pro-inflammatory mediators, indicating a direct cellular level effect. Clinically, sternal wound patients undergoing debridement with low-frequency ultrasound combined with negative pressure wound therapy, VAC, showed improved outcomes, including shorter hospital stays and faster wound closure compared to VAC alone. However, it's important for professionals to note that there are still relatively few high-level evidence studies, and recruitment issues have hampered some randomized controlled trials in this area, particularly for common conditions like diabetic foot ulcers.
And finally, vibration therapy is an emerging area with some intriguing results, shaking the wound better.
Vibration therapy is indeed quite new in wound healing. It has previously been found to promote angiogenesis, the growth of new blood vessels, in bone, and low-magnitude, high-frequency vibration therapy was shown to recruit mesenchymal stem cells to fracture sites in osteoporotic rats, accelerating bone repair. High-frequency acceleration has also been shown to preserve bone after tooth extraction in animal models. In a mouse wound healing model, low-intensity vibration increased angiogenesis and granulation tissue formation and led to higher levels of crucial growth factors like IGF-1 and VEGF. In humans, whole-body vibration has been shown to reduce pain in extremity, burn patients during and after therapy, and to mitigate bone loss in pediatric burn patients. While most studies in wound healing specifically are currently underpowered or inconclusive, and level-eyed data, the highest standard of evidence, is still sparse for electrical stimulation and therapeutic ultrasound, electrical stimulation currently has the strongest clinical evidence. This is an active and exciting area of research, and much more work is needed to fully understand the efficacy and optimal clinical utilization of these innovative new modalities.
Wow, we just covered an incredible amount of ground, truly diving deep into the intricate choreography of wound healing. From the immediate vasoconstriction and intricate cellular responses to the long-term remodeling and the countless cells, growth factors, and extracellular matrix components involved, it's clear this is one of the most complex and vital biological processes in our bodies.
What stands out most is that constant dynamic interplay between cells, growth factors, and the extracellular matrix that dictates every single step. And for professionals, understanding the significant challenges posed by chronic wounds, where that delicate balance is profoundly thrown off, is absolutely critical. But we also highlighted the exciting advancements, from sophisticated active skin substitutes to the incredible promise of gene and stem cell therapies, moving us towards more precise healing.
You now have a truly professional level understanding of this field, equipped with detailed insights into what well-informed truly means when it comes to how your body heals and how medical science is pushing those boundaries.
And as we wrap up, consider this provocative thought. Given the incredibly fine balance required for effective healing and the body's natural tendency towards a functional repair with scar formation over perfect regeneration, what might be the ultimate biological trade-offs that prevent adult humans from achieving scarless wound healing in all circumstances? What further profound biological secrets might unlock that ultimate regenerative potential, allowing us to heal like a fetus or even a salamander in every injury?
That's a profound question, and one that continues to drive innovation in this vital field, pushing us towards the future of medicine. Thank you for joining us on this deep dive into wound healing. We'll be back soon with another fascinating topic.